Welcome to my all-in-one CIE IGCSE biology video. I'm so excited that I've made this video. CIE is something that I've been determined to work on recently, and now I finally have time, so I really hope you guys find it helpful. So yes, CIE people only. Now, as always, my perfect answers pop up throughout the video, and they will be the wording that I think you should be including your answers in the exam to score maximum marks. Now, if you want me to do all the hard work for you, remember I have perfect answer revision guides available on my website, www.sciencewithhazel.co.uk, and they're available as a PDF, so go check that out. And there's also details about any revision courses I might be running. However, enough about that, let's get started. We're going to start by looking at the features that all living organisms have in common, and lots of people call that Mrs. Nurg or Mrs. Gren. So remember, if they say give some features that all living organisms share, you're going to say movement, respiration, sensitivity, you're going to say nutrition, excretion, reproduction and growth, and that just means getting bigger. So if it's non-living like a virus, you can easily say it does not move, it does not respire, it does not excrete. So just list any of the Mrs. Nurg factors and you will get the marks. Now, classification. There are billions of organisms that exist on our planet, and naming them has become a bit of a nightmare because the more species we discover, the more names they need to be assigned, and the whole thing gets into a huge mess, which is why we need classification systems. And this is just a way of classifying organisms into groups based on common features or features that they share. Now, the system we currently use is the binomial naming system, and that names all organisms according to two names, that is both the species and the genus. E.g. Mycobacterium tuberculosis, and that's the bacteria that causes TB. We do need to discuss what a species is just to make sure you understand that. And this is where groups of organisms can reproduce to produce fertile offspring, and this is the crucial thing. So dogs belong to the same species because they can breed and produce puppies, and these puppies should be able to breed with other puppies and produce fertile offspring, and so on and so forth. Remember what happens if a horse and a donkey reproduce? Well, they do reproduce to produce what's called a mule, but this does not count as a species because mules are sterile. That means that they're infertile, so two mules can't breed because they won't produce anything because of the fact that they're sterile. So a mule is not a species. So the traditional classification systems, so not ones based on the binomial method we use now, was based on evolutionary similarities, and it grouped organisms based on their morphology and anatomy, so effectively the bones which made them up. A more accurate way of classifying these organisms is to look at greater detail, so actually look at their structures, and that means looking at the sequence of bases in their DNA and how these sequence of bases determine which amino acids form which proteins. So that's a more accurate way of determining how we should group organisms. More similar sequences of DNA, so more similar amino acids and proteins, are more likely to be closely related. If we're going to use the correct nomenclature when we're talking about naming things, we can talk about the 5 kingdoms, and that consists of plants, animals, protoctists, bacteria and fungi. So what is a vertebrate? Well, it is an animal containing a backbone, a vertebra. So your spine is made up of vertebral column, so a vertebrate contains a backbone. Now, there are several groups of vertebrates you need to know about, and you need to know their distinguishing features. So for the mammals, their most notable points is that they have furry skin and they have mammary glands, which means they produce milk used to feed their offspring. Moving on to birds, they obviously have feathers, which you find on their wings, and then they have scales on their legs. With reptiles, so we're talking about lizards and snakes here, the most notable thing here is that they have dry, scaly skin. For fish, again, lots of scales here. They have fins which help them to swim. So they have gills where they filter water, and remember they have like a flap of skin which covers the gills, and we call that the operculum. And lastly, they have a lateral line containing sense organs. For amphibians, we're talking about things like frogs. They have moist skin. I've tried to bribe Lyra into sitting on my lap, so she's on a cushion. Princess Lyra. Has anyone seen that episode of Friends where Rachel brings in the cat without fur on a cushion? It's basically Lyra now. Look what I have to do to convince her to be my friend. She likes the cushion. So we know what a vertebrate is. It is an animal. No, Lyra. Oh, she's gone already. We know what a vertebrate is. It is an animal containing a backbone, so obviously an invertebrate is something which doesn't have a backbone. And now we're going to look at the arthropods, which is a group of animals without backbones, so they're all invertebrates, and they have what's called an exoskeleton, which means that their skeleton is found on the outside of their bodies, so things like crabs. Whereas as humans, we have endoskeletons because our skeletons are found within our bodies, because look, my bones aren't sticking out unless I've had a horrible accident. So starting by looking at the different arthropods, we're looking at the myriapods. They have antennae, which are the things that stick out. They have bodies made up of many segments, and they have a hard exoskeleton, so something like a millipede. When you're doing this, try and name the different numbers of wings, numbers of legs, that sort of thing, to help your answer plop out, and state whether they have antennae or not. So the insects now, well, obviously insects have antennae. They have bodies made out of 3 parts, which is the thorax, which is here, and we know this because our lungs are found within the thorax. The abdomen is further down, and then obviously at the top they have a head. They have compound eyes, 2 pairs of wings, 3 pairs of legs, so you'll see they have 6 legs. And the last thing to notice, which is weird, is that they have mouth parts. With arachnids, so spiders, they have simple eyes. They don't have compound eyes, so they can't see as well as things like flies. Their head and their thorax are combined. We know that spiders have 8 legs, which means they have 4 pairs of legs. They have powerful jaws because arachnids are predators. They kill and eat other animals. And they have and I love this word, they have a spinneret, which is the place where their spider silk comes out of. So the crustacean now, lots of people like eating these. We're talking about lobsters and crabs, for example. So what do you think of when you think of a lobster and a crab? Well, you think of their hard outer casing, which we actually call a carapace. They have claws with serrated edges, which is why you don't want to get your finger stuck because that could hurt. They obviously have eyes, they have jointed limbs, and lastly, you may not know this, they have gills under their shell. We now need to look at plants, so starting with flowering plants whose proper name is the angiosperms. Again, you just need to list a load of features. So first of all, they can produce flowers, they produce fruits and seeds, they have extensive root systems for obtaining water and mineral ions, and coupled with this is transport systems such as the xylem and phloem. The stomata allows carbon dioxide into the leaves and oxygen and water out. And lastly, they can be monocotyledons or dicotyledons, and that's to do with how many leaves they produce initially. Mono meaning 1, so a single leaf. Dicotyledons produce 2. Ferns are extremely different, even though they have leaves. We have to call the leaves fronds, and that means that they're long and thin. These fronds contain sporangia, and it's these sporangia that release spores, which is how the ferns reproduce. They also have what's called underground rhizomes. Now, this is because they have nodules, which are like small blobs which exist underground, and what these do is they send out roots and shoots, helping our fern to reproduce. They also have very simple root systems, so compared with the angiosperms, which have extensive root systems, we say that ferns have simple, true root systems. Lastly, viruses, nice and simple here, just state that they consist of a protein coat which surrounds genetic materials such as DNA or RNA. Right, we now need to go over the dichotomous keys part of the specification. Now, I think the easiest way here is to show you past exam question to work out how you're going to approach it, because really I can't just explain it without an example. So figure 1. Figure 1.1 shows 5 different insects. Yeah, cool, those insects are really nicely drawn actually. Use the key to identify the insects in figure 1.1 and write the letter for each insect in table 1.1. So the easiest way to approach this is from the animal point of view, not the table. So that is my first recommendation. So I'm going to look at insect A first of all. You can see it's quite squat, it's short and fat. It has 2 antennae poking out the top, which are here, so make sure you know that those are antennae. And it has a striped casing. So now let's have a look at how A fits into this table. So 1A, body is long and thin. That definitely doesn't apply to insect A, so I'm going to go to B. Body is short and rounded. Yes, it is short and rounded. So as the table tells me, I'm going to go to number 3. So number 3A, no visible antennae. Well, I can see the antennae, I've marked them off. So we go to B, visible antennae, which tells us to go to step 4. Step 4, body has a striped pattern, or body has a dotted pattern. I think we can safely say A has a striped pattern, which means that A is, and excuse my pronunciation, Graphosoma linatum. So I'm going to put A in here. Let's go to B now. So B looks like a ladybird actually. Has very small antennae. It has a small rounded body with spots. So 1, Body is long and thin. No. Body is short and rounded. Yes. Go to 3. No visible antennae. That's not true. We can see it has antennae. So B is correct. It has visible antennae. So we've been told to go to 4. Body has a stripe pattern. No. And we know that belongs to A anyway. Body has a dotted pattern. Yes. So B needs to be written here. Looking at C, we've got a fairly rounded body. Not as rounded as the first two, but fairly. We can't see any antennae, and it seems to have quite a uniform black colour. So, going in at 1, body is long and thin. No. Body is short and rounded. Yes, so we're going to go to 3. 3, no visible antennae. I think we can safely say it has no visible antennae, so C goes in here, Copris lunaris. Moving on to D, so it's long and skinny, it has some blobs on it, it has antennae. So 1A, body is long and thin. Yes, that's true. So we need to go to 2. 2A, body has a spotted pattern. It does have a spotted pattern, which is why Alauus oculatus is D. And then never just assume, you need to work it out properly and make sure that you agree. Don't just put E in as that missing gap. So E, we can see it's long and thin, it's grey, it has antennae. So we go in at 1, its body is long and thin, yes, so we're going to go to 2. Body has a plain pattern. I think it's safe to say it is plain, which is why I'm going to put E there. Now we're going to look at the plant and animal cell, very basic biology here. First of all, let's start by listing the organelles that both animal and plant cells share. So remember, they both have cell membranes, cytoplasm, nuclei or nucleus. They have ribosomes, mitochondria. Now, in terms of the plant cell, there's a few extra organelles you need to list. That's the cell wall, the vacuole, and also chloroplasts. We now need to look at the role of each of those organelles in turns. So because it's so key that you get these really basic questions right in your exam, because if they say, what does the nucleus do, you need to be able to write that. So what does the nucleus do? It controls the activities of the cell. What does the cytoplasm do? It's where chemical reactions take place. What is the role of the cell membrane? It controls what enters and leaves the cell. What is the role of ribosomes? It's where protein synthesis takes place, i.e. it's where proteins are made. So I've already touched on lots of different organelles which you find. We just need to add a couple more. So I said that ribosomes make proteins. Now these ribosomes may be attached to something called the rough endoplasmic reticulum. The reason why we say it's rough is if you look at it under a microscope, it has small balls on it, and that's the ribosomes, hence rough. Now they're obviously, if you ask the role of the rough endoplasmic reticulum, clearly it is in protein synthesis. Which organisms do not contain mitochondria or rough endoplasmic reticulum? Well, these will be the prokaryotes. So remember, the prokaryotes do not have a distinct nucleus. So things like the bacteria or viruses. Looking more closely at the plant cell now, what is the role of the cell wall? First of all, state that the cell wall is made out of cellulose and that it protects and supports the cell. The vacuole, remember, that's filled with cell sap, which also helps to maintain the structure of the cell. Lastly, chloroplasts, remember they're full of a green pigment called chlorophyll. It gives leaves the green colour, and that's where photosynthesis takes place. A couple of tricky words you may have come across is eukaryotes and prokaryotes. Don't worry, it's just a very posh way of describing the type of cell we're talking about. Eukaryotes are all animal cells as we know it, and that's because they contain membrane-bound organelles such as nuclei, mitochondria, etc. So an animal cell, like I said, is an example of a eukaryotic cell. Prokaryotes, we're talking about viruses and bacteria here because they contain no membrane-bound organelles, so they contain no nuclei, which we know because they contain strands of DNA or RNA instead. So we now need to take lots of different types of cell in turn and know quite a lot of information about them. So I'm going to start with the bacterial cell. We can see from the diagram that a bacterial cell has a cell wall. Sometimes it has a slime capsule around the edge. Sometimes it has a flagella, which is a tail that helps the bacteria to move. As I've already said, it doesn't have a distinct nucleus. Instead, it has a circular chromosome, which we call a nucleoid. It has other small rings of genetic material. We call these plasmids, and that, remember, is important when we talk about genetic engineering. Then you find more typical things such as cytoplasm and cell membranes. In terms of things like whether they're pathogenic or non-pathogenic, remember that they can be both. So a pathogen is a microorganism that causes disease. It makes sense, therefore, that some bacteria are pathogenic. Such examples include pneumococcus, which is responsible for pneumonia, tuberculosis, remember that gives people TB where they cough up blood, very horrible disease. However, some bacteria are very useful, like those used in yogurt making. The example here is Lactobacillus bulgaricus. Remember lastly that bacteria are unicellular, which means that they're made of one cell only. Looking at viruses now, because that leads on quite nicely from bacteria, these are very, very small things. They're much smaller than bacteria. They're far more simple because they're simply made out of a protein coat which surrounds either DNA or RNA. They don't have any of the typical organelles you would find in other types of cell. The crucial thing here, as I've already said, is that they're non-living. They do not excrete, they do not respire, they do not grow. They're always pathogenic. There's no such thing as a good virus. They're always out to hurt you. And examples here include the flu virus, the cold virus, HIV, which is very famous because it causes AIDS. Next up, we're looking at the protoctists. This is known as the dustbin kingdom. Lots of various organisms which don't fit into the other categories fit into protoctists. Some of them have animal cell properties, some of them have plant cell properties. Starting with algae and also Corella. These both have chloroplasts, which means they're more plant-like. Things like amoeba are more animal-like. You'll see that they don't have chloroplasts, they don't have a cell wall as such. And they all use diffusion in order to obtain their nutrients and get their oxygen. One key one you need to know about is Plasmodium. This is pathogenic because it causes the disease malaria. And the Plasmodium is the small protoctist that lives in the female mosquito's bodies, and that's what she injects when she bites you. So that's actually what gives you malaria. Do note that they can either be unicellular or multicellular, so made up of one cell or many cells. Fungi now. Now fungi, they're quite easy because you can draw literally a plant cell but just make it slightly more circular. So it has the same organelles you would find in a plant cell, apart from the fact it obviously doesn't have chloroplasts, but it does have a cell wall. This is made out of chitin, you do need to know that. It has a cell membrane, cytoplasm, it has a vacuole. Now, there are lots of different examples of fungi, including mucor and mushrooms. Do notice that fungi carry out saprotrophic nutrition, and that means that they extracellularly secrete enzymes onto dead matter, which it breaks down and then absorbs as its food. Crucial words here are extracellularly secreting enzymes which break down dead matter, and that's how they actually obtain their food. There are some useful examples of fungi, including yeast. Now remember, that's used in beer and bread making. Why? Because when yeast undergoes anaerobic respiration, that means respiration without oxygen, it breaks down glucose into ethanol, which is clearly used in beer making, and also carbon dioxide. And it's those bubbles of carbon dioxide that actually help that bread to rise. Now we're going to look at organisation within organisms. So we're looking at key definitions. The crucial thing here is to just learn one definition and then just use it as a template for all your other definitions. So I'll explain what I'm talking about right now. So if we start, we look at a cell. Now remember, cells are full of organelles, which I've just listed, nucleus, cell membrane, etc. So what is a cell? Well, it's a group of organelles working together to perform the same function. We're going up a step further, so we're now looking at tissue. What is a tissue? It's a group of cells working together to perform the same function. Then we're going to look at organs. So what is an organ? It's a group of tissues working together to perform the same function. And lastly, what is an organ system? It's a group of organs working together to perform the same function. And lastly, what is an organism? Well, it's a group of organ systems working together to perform the same function. Now I need to list all the various organ systems within the body, which I've always been really bad at remembering all of them. So I'm going to try. There's the digestive system, there's the endocrine system, reproductive system, circulatory system, respiratory system, nervous system, and excretory system. But if we focus in on the digestive system, for example, we could first of all take the fact that the digestive system is obviously the system. So what organs make up that digestive system, that organ system? Well, it's things like the stomach, esophagus, pancreas, small intestine, large intestine. Then we can look at the tissues. So for example, in the stomach, you've got glandular tissue, which secretes hydrochloric acid. You've got muscular tissue which helps churn the food. Don't worry too much about this detail, by the way. I just want you to get the full picture so you're not thrown off if a question's slightly strange in the exam. And then you can obviously look at the cellular level within the stomach and the organelles. So it's a big hierarchy really. So specialised cells, this is a nice topic. I would get into the habit of drawing out each cell and then labelling it, and then hopefully it'll be easier to learn than just a list of separate points. So starting with the red blood cell, clearly that transports oxygen around the body. It does this because it contains a protein pigment called hemoglobin, which binds to the oxygen, forming oxyhemoglobin. In order to improve its efficiency, it has a very distinct shape. So it is a biconcave disc shape, which means it has an increased surface area to volume ratio, so it can transport more oxygen, and it has no nucleus to allow for more hemoglobin. It's also extremely flexible, allowing it to pass through the smallest spaces and along the narrowest of capillaries. The muscle cell, this is adapted so that it can contract and relax in order to move the muscles. Cells here are very long and contain protein fibres, and the protein fibres can shorten when there is energy available, which means that the muscle contracts. The ciliated cell we find lining the trachea, so we're looking at our respiratory system, and remember the trachea is actually the windpipe. So the ciliated cell contains cilia. These are small hairs which waft, and what they do is they waft mucus laden with bacteria out of the trachea into the mouth, where it can be swallowed and destroyed by the hydrochloric acid in the stomach. And this is a really good defense system against microorganisms. The motor nerve cell, remember there are lots of different nerve cells. This is the motor, so it transports signals from the central nervous system to the effector. So its real role is to conduct electrical impulses. The long part of that motor cell is called the axon, and that carries the electrical impulse. Don't forget that it is surrounded by a fatty sheath, which we call the myelin sheath, and that increases the speed at which the impulses may be conducted. They have lots of branched endings so that they can reach lots of other cells. The sperm cell now, so remember these are made by the testes of males, so the whole point of a sperm cell is reproduction. Remember, it fuses with the ovum in fertilization to produce a zygote, so its real role is reproduction. Now it has to swim through the vagina, through the cervix, through the uterus to the oviduct where fertilization takes place, and it has a flagellum, a tail, to enable this. It has a whiplash tail to help it to swim. Its middle section contains many mitochondria, which makes sense because they release lots of energy required to power that sperm on its long journey. In its head it contains an acrosome, which is full of digestive enzymes which help to digest the outer layer of that egg so it can actually penetrate it. And don't forget that its nucleus is haploid, which means it contains only one set of chromosomes, which makes sense because the ovum also contains only one set of chromosomes. So when they fuse at fertilization, a diploid cell is made, which makes sense because we want our zygote to be diploid because it will then go on and become our baby. So the ovum now, we've already said it's involved in reproduction. We've said it contains a haploid number of chromosomes in its nucleus. The only thing to notice here that it has an outer layer which is like a jelly coat that changes once the sperm has penetrated through. Moving on to plants now, so the root hair cell has a very distinctive shape. Why? Because remember its primary role is to absorb water and mineral ions, so it has a root hair which will increase the surface area for absorption, and this absorption occurs by osmosis in terms of water, and mineral ions are absorbed by active transport. I've already touched on the xylem and the phloem. Let's look at them in more detail now. So the xylem transports water, and it transports the water from the roots up the plant to the leaves, to the shoots where it is required. Now it is made out of dead cells which form a continuous column. The end of the cells has broken down, so there's no cytoplasm, and that means you effectively have a drain pipe to transport that water. Do notice that its walls are made out of lignin, and that helps to strengthen the xylem and stop it collapsing. The phloem transports sugars. When we look at the structure of the leaf and we look at the layers, we know that there is the palisade layer, which is the palisade mesophyll, and these are what your regular plant cells look like. So do remember that they just contain lots of chloroplasts which absorb sunlight for photosynthesis, which means they make sugar, or they make glucose for the plant.
So lots of people struggle with converting units, and I wanted to give you a really straightforward way of doing this. So let's have a look down here. We've got lots of different scientifically approved units. So we've got the picometer, which most of you won't come across, but I've just added it there for completeness sake. Then we have the nanometer here, which is far more likely to come up. Micrometer, millimeter, which you'll be familiar with because you'll have that on your rulers. meter, which is obviously much larger than that. You might have wooden meter-long rulers in your science lab, but you won't really come across them apart from there. Kilometers, that's very straightforward because we know kilometers. How many kilometers is it to the beach? 5. And then lastly, megameter here, which you probably haven't come across. So what I'm trying to show you here is you can use this simple expedient of timesing by 1,000 to convert between the various steps. So to get from millimeters to micrometers, simply times by 1,000. To get from micrometers to nanometers, times by 1,000. It obviously works the other way around. If you need to get from nanometers here to micrometers here, divide by 1,000. But I didn't want to make this diagram too complicated, so as long as you know what you're doing, you can obviously reverse it. And I'm going to show you some examples right now. So let's start by converting 5 meters to micrometers. So let's have a look in our table, and we need to times by 1,000 twice. And then just write out your answer now. So it becomes 5 million micrometers. We're now converting 3 millimeters to micrometers. So we're going from here to here, so we just need to times by 1,000. So we're just timesing by 1,000 once. Next up, converting 10 centimeters to nanometers. This is more complicated because we've got centimeters, which isn't included here. So let's first of all convert centimeters to millimeters. Hopefully that's really straightforward for you because again, you have a ruler. It says 10 centimeters on it, and you'll be able to work out from your ruler that 1 centimeter is 10 millimeters. So therefore 10 centimeters is going to be 100 millimeters. And then we're just looking back at our little table thing, and we need to see how we're getting from millimeters to nanometers, and we're timesing by 1,000 twice. And let's just add some little commas to try and make that make more sense. So that's 100 million nanometers. And now we're going to go the other way. So we're converting 22 micrometers to millimeters. So this time we need to divide by 1,000. So 22 micrometers is 0.022 millimeters. So now we're going to practice what we've just learned, and we're going to start by looking at question 1 on the right hand side. The diagram below shows the general structure of an animal cell as seen under an electron microscope. Calculate the magnification of the image and show your working. So let's start by writing our formula triangle out. That does not look like a triangle. So it's I am, which means that magnification equals the image size over the actual size. The easiest way to do this is not actually measure the diameter of the cell. You need to use this scale bar here. We can see from the scale bar that its size is 5 micrometers. So we're going to put that there as the actual size. In terms of the image size, you're going to use your ruler to measure the length of that line here. I know it's hard because I'm doing it on the iPad, but let me tell you that it is 17 millimeters. Now the important thing with these calculations is to get all the units being the same. So remember, I just taught you how to convert, so if you actually look back in the video you could see this. But to get from millimeters to micrometers, remember you times by 1,000. So let's convert 17 millimeters to micrometers by adding 3 noughts, divide it by 5, and you get a magnification which is 3,400 times as big. The photograph shows part of the cytoplasm of a cell. The magnification of this image is 200,000. Calculate the actual width of the organelle X. Show your working. So as always, using our triangle I am, we're looking for the actual length, so that's going to be image size over magnification. So let's substitute in the magnification first of all, because that's nice and straightforward. You're going to use your ruler now to work out the width. It's the width, not the length of organelle X. So do that in millimeters and you'll get an answer which is 20 millimeters. I like to keep my units in so I know what's going on. We're converting that to micrometers as per usual, so that becomes 20,000 divided by 200,000, giving us a value which is 0.1 micrometers. Now we're moving on to transport, so let's first of all touch on the 3 types of transport. So we're looking at diffusion, osmosis and active transport. So you need to know the definitions of these terms in great amount of detail. Now remember, with diffusion, it's the net movement of particles
from an area of high concentration to an area of low concentration. So that's the reason why you spray perfume in one side of the room where there's a high concentration of perfume particles, and it moves across to the other side of the room where you can smell it. That is by diffusion. It is a passive process. It does not require energy. Osmosis is very similar to diffusion, however it involves the movement of water, which is why our definition this time is osmosis is the net movement of water from an area of high water potential to an area of low water potential across a partially permeable membrane. Now, potential is just a really posh way of saying concentration. So somewhere where there's lots of water to somewhere where there's little water. And do add that it's across a partially permeable membrane. If there's no partially permeable membrane, it's not osmosis, it's just diffusion. So notice when water leaves the stomata from that leaf, that is by diffusion because stomata is a whole, not a partially permeable membrane. Lastly, active transport. As the name suggests, it's an active process. This means it requires energy. The reason being is because it's the net movement from an area of low concentration to an area of high concentration, so against the concentration gradient. So we need to go into slightly more detail about how active transport actually works. So remember that there are carrier proteins within the membrane. These carrier proteins recognise the particle to be transported. The particle is transported across the membrane using the carrier protein, and this process requires energy from ATP. Particles then obviously released into the cell, and lastly the carrier protein returns to its original position. Let's touch on amoeba now. Remember, the amoeba is an example of a protoctist. This is a single-celled organism which can use diffusion in order to obtain all the nutrients it needs. So oxygen diffuses from outside that amoeba into the amoeba from an area of high concentration surrounding the amoeba to a low concentration inside the amoeba. The reason why diffusion is appropriate is because it occurs very quickly, because the amoeba is single-celled, which means it has a large surface area to volume ratio, and therefore the speed of diffusion is fast enough to allow oxygen in as and when it is required. Larger organisms, which are multicellular, have a much smaller surface area to volume ratio. Diffusion is not suitable, it is too slow, which is why they develop the need for a circulatory system. So we need to look at biological molecules, starting with organic compounds, and organic means relating to the element carbon, and we're including in our organic compounds carbohydrates, lipids and proteins. So starting with carbohydrates, look at the name to work out which elements exist within it. So carbo meaning containing carbon, hydrated meaning containing the elements found in water, so they contain hydrogen and oxygen also. Proteins also contain the elements nitrogen and sulfur, and nucleic acids contain the elements phosphorus and also sulfur. When we look at carbohydrates, we can divide them broadly into two groups. These are monosaccharides and polysaccharides. Polysaccharides contain many sugars. Key examples here include cellulose, which is the sugar found in cell walls, and also starch, which remember is the storage compound found within plants, and glycogen, which is found within animals and fungi. So looking at proteins now, so what is a protein? Well, it's a chain of amino acids whose order has been determined by genes. This sequence of amino acids determines the protein shape, and the protein shape and structure give the protein its particular function. So for example, amino acids found within keratin, which is a protein, give its very structural support function, whereas amino acids found in hemoglobin give it a very different role, which is to do with the transport of oxygen. So how does the structure of an antibody allow it to work? Remember, antibodies are made from white blood cells, and they're involved with fighting pathogens and the immune response. So remember that each antibody has a specific binding site. The binding site has a specific shape, and therefore this can recognize very specific molecules. And remember, molecules which bind to the antibodies are known as antigens. Enzymes are key proteins, which we need to know lots about. And an enzyme is a biological catalyst. This means that it speeds up the rate of a chemical reaction without being used up. Now, an enzyme has a very special part on it called an active site, and that's the biologically active part of the molecule. And what happens is the substrate molecule binds to that active site, it forms an enzyme substrate complex, which then splits up to form the useful product that we're after. Now we need to just touch on enzyme activity. So the two things you can alter is both the temperature and the pH. So if you look at this graph, you can see at low temperatures, the enzyme activity is low. The reason for this is all to do with chemistry, it's to do with collision theory, because at low temperatures, enzymes have very little kinetic energy, as do the substrate molecules, so it means that the enzymes and the substrate aren't coming into contact very often. That means they obviously can't bind at the active site, and so the reaction can't be catalyzed. As we increase the temperature, you can see the enzyme activity increases. That's because those molecules are coming together more often, and at 37 degrees, as is the case with most animals, you will find that enzyme activity has reached its peak. It is at its optimum temperature, the best temperature, and that means the enzymes and the substrates are coming together very frequently. After this temperature and above this temperature, we see a massive decrease in enzyme activity, and that's because the enzyme has become denatured. Never say the word killed. They're not living, they can't be killed. You need to say they're denatured. And what that means is that the enzyme's active site has changed shape, meaning that the substrate can no longer fit. If we take a look at pH now, you can see a very distinctive graph shape here, and that's because enzymes have different optimum pHs. And if the pH is either too high or too low, around that optimum pH, you'll find that the enzyme denatures, which is why you have that cone shape. Because let's look at the enzyme which has an optimum pH of 7. If you go to 6.5 or 7.5, the enzyme will denature. And this is true for all enzymes. Some enzymes prefer different pHs to other ones. So proteases in the stomach, for example, because they're surrounded by hydrochloric acid, they'll have an optimum pH of approximately 3, whereas throughout the rest of the digestive system, you find a slightly alkaline optimum pH, so of around 8, which is why you can't take stomach protease and put it in the small intestine and expect it to be okay. Right, we're going to be talking about all things to do with plants, starting with that key plant process, photosynthesis. Remember, photosynthesis is carried out in the chloroplast of plant cells. They contain chlorophyll, which absorbs that sunlight and actually carries out the process of photosynthesis. And this is the method by which green plants make their own food. So let's start by looking at the word equation for photosynthesis. Remember that it's carbon dioxide plus water forms glucose and oxygen. This is why it's such a great process, because of the huge volume of oxygen photosynthesis releases. In terms of the balanced symbol equation, try and learn this off by heart and remember that 6 is very important. So you've got 6 CO2 plus 6 H2O forms C6H12O6 plus 6 O2. Now, unfortunately, there are lots of different factors which can act to reduce or limit the rate of photosynthesis, and we call these limiting factors. You need to know the definition of a limiting factor. It's a factor which in a reaction is in the shortest supply, and a lack of this factor is the reason why a rate of reaction no longer increases. Now, in terms of photosynthesis, there are 3 limiting factors you need to know about. These are carbon dioxide, light, intensity and temperature. So any one of these may act to limit how much photosynthesis can take place, and we're going to talk about each of these in turn in a different situation. So let's think about early morning. We have a green plant and it's trying to photosynthesize in early morning, but what could be limiting how much photosynthesis takes place? Well, it's early morning, so it's quite cold, so obviously temperature is going to be a limiting factor here. Another limiting factor will most likely be light intensity, because it's not as light early morning as it is at midday. So despite carbon dioxide levels increasing, you will find that low light levels, low temperatures limit the rate of photosynthesis. The reason being is that in the morning, low temperatures obviously mean low kinetic energy, so all those enzymes involved in those chemical reactions involved in photosynthesis can't actually come together, they don't collide as frequently with their substrate molecules, and that's all because of little kinetic energy. If we take midday now, we know that the temperature will have increased, we know that the light levels will increase, so neither of these things will be limiting factors. The most likely limiting factor at midday will be carbon dioxide levels. And be sure that you can look at graphs on all of these factors and make sure you understand what is going on. Let's quickly look at the role of diffusion in gas exchange in plants.
So remember that carbon dioxide is needed by plants in photosynthesis, so it will diffuse into the leaf through the stomata. Oxygen is released by photosynthesis, so that will diffuse out of the leaf, also by diffusion. However, don't forget that plants are living organisms, so that they respire, which is a bit confusing, which means they need some oxygen, obviously, in order to carry out respiration. So if you look at it over the course of the day, you will find that during the night, because the plant can't photosynthesize, because obviously there's no light, it will just be respiring, which means that more oxygen will enter the leaf than will exit the leaf. During the day, photosynthesis tends to occur at a faster rate than respiration, so in this case, more oxygen will leave the leaf compared with diffusing into the leaf. So, we know photosynthesis is the method by which plants make food, and they make that in the form of glucose. So what do they do with that glucose? Well, remember that glucose contains carbon, hydrogen, oxygen, so it obviously contains components that can be used to make other biological molecules, and this includes fats and proteins, so that glucose is used to make fats and proteins. It's used to make storage compounds, such as starch, which the plant can call upon in lean times, and it's also used to make the sugar cellulose, which is an important component of plant cell walls because it gives it its structural integrity. Carrying on with the plant theme, we're now going to look at the structure of a leaf and how it is adapted for photosynthesis. Let's make some generic comments at the beginning by stating that a leaf has a large surface area, which is obvious, so that it can absorb more light. It's thin, so the gases don't have to diffuse too far. Looking very closely now at the structure of the leaf, you need to be able to label all those layers and discuss which each of them do, and you need to be very specific here. So we're going to start with the waxy cuticle. The waxy cuticle is there to prevent transpiration, and remember that transpiration is the loss of water from the leaf. So a nice thick waxy cuticle prevents excess water loss. Next up we have the upper epidermis. Remember, this is transparent and it allows light to enter the leaf. You don't need to say anything more than that. The layer beneath this is the palisade mesophyll. Mesophyll is just a fancy way of saying tissue. The palisade mesophyll is what your generic plant cell looks like. So it's your rectangle block. It's crammed full of chloroplasts, so it contains lots of chloroplasts for photosynthesis, and this is where photosynthesis takes place. Under this you have the spongy mesophyll. The important thing to note here is the presence of plenty of air spaces which allow gases such as carbon dioxide and oxygen to diffuse. You'll also find the vein here, and the vein contains the xylem and the phloem. The xylem brings water into the leaf, the phloem transports sugar away from the leaf. Then we have the lower epidermis. Not a lot to say here. The next layer is the most important layer, and that contains the guard cells and the stomata. Now the guard cells control whether the stomata are open or closed, and the stomata allow carbon dioxide into the leaf and oxygen and water to leave the leaf. Relating to the plant mineral ions, luckily you don't need to know too much, just learn the role of nitrates and magnesium. So the mineral ions are present in the soil around the roots of the plant, and the plant obviously needs them to be healthy. So it absorbs both magnesium and nitrates by active transport, so against the concentration gradient, and it uses the nitrates to build proteins, and it uses the magnesium to manufacture the chlorophyll found in chloroplasts. You need to know some deficiency signs. So with magnesium, clearly you won't be able to manufacture chloroplasts or chlorophyll anymore, so you see yellow leaves, and if you've got a lack of nitrates you will see a stunted, poorly grown plant, so it's very short.
Balanced diet now. So what is a balanced diet? Well, it's one which provides all the nutrients in the correct proportions which are needed to carry out life processes.
Moving on to humans now, we're looking at balanced diet, so we need to look at the various nutrients that you need for a balanced diet, their roles within the body, the foods that they're found in, and any deficiency diseases. So let's have a look and start by listing these nutrients. So we're looking at carbohydrates, fats, proteins, minerals, vitamins, water and fibre. I hope I've listed them all. So starting with carbohydrates. Foods which contain lots of carbohydrates include things like bread and rice and pasta. Carbohydrates are an important source of energy. Proteins now you find lots of protein in meat such as chicken and beef. Protein is important for the growth and repair of muscles.
Now remember, lots of people take protein shakes to the gym. Why? Because they're trying to build their muscles at the same time, so they like to take a source of protein. So try and remember it from that point of view. If you have a lack of protein, you get a really horrible disease called kwashiorkor. People are seeing very distinctive ball-like stomachs where your stomach comes out super far, and that's a symptom of kwashiorkor. Fats, foods which contain a lot of fat include dairy foods such as butter and cream. I'm just giving you a few examples, it's not exhaustive. Now fats are a very concentrated source of energy and they provide insulation, i.e. they help to keep you warm. Moving on to some specific examples of vitamins. So vitamin C, you find this in citrus fruits such as oranges and lemons. Vitamin C is important for the repair of tissues, so it helps to stick together the cells in the lining of your mouth. And if you don't have enough vitamin C, you get a disease called scurvy, which was infamous in the 1500s when sailors used to go out to sea. They never got enough oranges and lemons, and then you'd see very characteristic mouth bleeds. So scurvy is the deficiency disease, and the way to get rid of that is by eating lots of oranges and lemons. Vitamin D now vitamin D is important for strong bones. You find it in fish liver oils, which is gross, I hate cod liver oil, but it's also manufactured by the action of the sun on the skin, so you can get a lot of vitamin D sunbathing, but obviously not too much, don't get burnt, that's not a good idea either. And lack of vitamin D leads to rickets in children. Now we're going to move on to minerals such as iron. Iron is found in red meat and spinach. It's important for healthy blood. It's a really important component of hemoglobin, which is found in red blood cells. Lack of iron leads to anemia, which is when you feel really tired and exhausted all the time. And lastly, calcium. Calcium is a mineral which is important for strong teeth and bones. It's found in dairy products such as milk. Lack of calcium will also lead to rickets. Last two things, fibre. So fibre is essential to help food move through your digestive system. Without fibre, you're liable to get constipation. Vegetables and fruit contain a lot of fibre. And water we know we need to survive. Without it, we would die very quickly, and that's because it supports all the chemical reactions that take place in our bodies. And just be aware of a deficiency in energy and protein is known as marasmus. And that's when you see muscle wasting on limbs. Now, an important side note is to notice that we need a balanced diet full of all these nutrients in order to stay healthy. But obviously our requirements will vary depending on our age. So older people will need less food, less of each of these nutrients compared with teenagers, for example. Pregnant women, they'll need more because they're supporting the growing fetus. Teenage girls will need more iron because they've started their periods. People who exercise will need more protein to help them with the growth and repair of their muscles. And lastly, pregnant women will need more dietary energy compared with people who are not pregnant. So it's a bit of a common sense part of the specification, but just be aware that there are differing requirements. Now we need to look at digestion. Let's first of all discuss the definition of digestion, which is that it's the breakdown of large insoluble molecules into small soluble ones. The reason being that we need to take our food into our mouth and we need to break it down into teeny tiny pieces, change its structure so that it can be absorbed through the walls of the small intestine. So that's what we're on about when we're talking about digestion. So there are various enzymes you need to be aware of. Firstly, amylase, and notice that enzymes tend to end in ASE, so A's. Amylase is made in the salivary glands in your small intestine and your pancreas, and what amylase does is it catalyzes the breakdown of starch into glucose. Now we need to look at protease. This is more straightforward because, as the name suggests, it breaks down proteins. So protein is the substrate and it breaks down proteins into amino acids. These are the products, and protease is found in your stomach, in your small intestine and your pancreas. Slightly more detail on protease enzymes then. So we know these break down proteins. However, there are two you need to know the name of, and that is pepsin and trypsin. Pepsin is simply the protease found within the stomach, whereas trypsin is found within the small intestine. The last enzyme you need to be aware of is lipase. Lipase breaks down lipids, or fats as their more colloquial name, and lipids are broken down into fatty acids and glycerol. So do try and remember that's the most complicated one. Now, I've already told you about chemical digestion, which relates to enzymes entirely because that's totally altering the structure of the food molecule. We need to also look at mechanical digestion, which is a far more physical process that involves just chopping that food up into smaller pieces, but doesn't alter the structure of that food. So if we think about where mechanical digestion takes place, it will be in the mouth, your teeth chew. It'll also be in your stomach, where your muscular walls churn that food up and break it up into smaller chunks. So the teeth, right, we need to be able to name all the teeth and talk about what they do. Remember, the teeth are very important in mechanical digestion. They actually alter the size of the food to make it more easily digestible by enzymes. So let's name our various teeth. So the front teeth here are known as the incisors. Their job is simply to cut or slice food. The canines, which in humans has become hugely reduced, but in animals like dogs you see that they're very long and pointed. This grips the food, so it holds it in place. Then you have larger teeth behind that, which we call the premolars. Their job is to crush and chew the food, and then the larger teeth at the back, they're the molars. They provide the grinding surface to actually grind that food up. So what causes tooth decay? So very painful when we have cavities. Now that's all to do with the bacteria, because obviously the bacteria feed on any excess food in your mouth, which is why it's so important that you clean your teeth properly. So the bacteria stick to your teeth and they make that plaque, which the dentist can sometimes scrape off. The bacteria respire, and while they're doing this they produce an acid, and it's actually that acid which erodes away your teeth, so particularly the enamel and the dentine of your teeth. So how can we actually protect our teeth? Well, clearly we need to eat a diet low in sugar, because we know sugar speeds up the rate of decay. We need to brush our teeth regularly, and ideally after a meal rinse to remove any excess food that might be stuck between our teeth. And I've even read that you can eat crunchy vegetables because they effectively help remove that food too. So we'll start at the mouth. So I've already said we've got physical digestion from the teeth. Chemical digestion comes from amylase, which digests starch into glucose. The food then passes down the food pipe, which we're going to have to call the esophagus, and peristalsis is a process whereby the muscular contractions of the esophageal wall force that bolus, that ball of food, down into your stomach. Here, muscular contractions of the stomach lining help churn the food. You've got the secretion of hydrochloric acid. This has two jobs. It breaks down the food, and it also destroys pathogens, and that's the reason why you don't get sick all the time if you eat some slightly dodgy food. Sometimes the food you've eaten is so dodgy that you still get sick, but for the most part you'll find your stomach acid will destroy those pathogens. Remember, a pathogen is a microorganism which causes disease. Protease is secreted, that breaks down proteins into amino acids. At this point, the stomach empties and the food flows into the small intestine. Well, there'll be further peristaltic contractions. I don't know if that's the exact version of the word peristalsis, but it's similar enough, which force the food along. We've got more enzymes being added here, lipase, protease and amylase, and note that they'll have a differing optimum pH from the protease in the stomach. They'll have a much higher optimum pH. We need to mention bile now. Now, bile is an interesting substance. I think it's green. It's made in the liver, you must remember that. It's stored in the gallbladder and it's released into the small intestine. And it has two main jobs. First of all, bile is an emulsifier, and what that means is that it breaks up large fat droplets into smaller fat droplets. Why? Because it creates a much larger surface area, because small droplets have a much larger surface area than large droplets. And that means that lipase can work more easily on the lipid molecules, on the fat molecules. But if you can't bother to write all that, just say that it emulsifies. Its second role is to neutralize stomach acid. It brings up the pH to an alkaline pH, approximately 7 or 8. And what that does is it means that those enzymes that have been released into the small intestine don't get denatured by all the acidic food coming along. So that's bile's two main jobs, to emulsify and to neutralize. So at this point we've done lots of digesting. Our food molecules are very small, they're very soluble, which means they can now pass through the walls of the small intestine into the bloodstream. They often ask you what the adaptations are of the small intestine lining, so you're going to talk about villi primarily. So remember, villi are these structures shaped like this, and they provide a very large surface area for absorption, and that's also maximised by the presence of microvilli. This video is so detailed it's making me lose my voice. So yeah, microvilli help increase the surface area further. You've got a short diffusion distance. you've got a plentiful supply of blood capillaries, and you've also got the presence of lacteals, which are for fat absorption. So the small intestine is extremely adapted for its role. Going into more detail to do with the microvillus therefore, so we know it increases the surface area for absorption. The epithelium is one cell thick, which provides a short diffusion pathway. Goblet cells produce mucus, like in the trachea, and what this does is protects the lining of the small intestine. Good blood supply from within the microvilli to transport those digested foodstuffs away from the small intestine to elsewhere in the body. And lastly, the lacteal transports fatty acids and glycerol. So once all that food has been absorbed and you've just got the leftover undigestible food, it passes into the large intestine, and here water is reabsorbed into the blood. Lastly, the feces, because we're now at the feces stage, that's the fancy word for poo. The feces are stored in the rectum before they pass out of your body via the anus. We call this egestion, the removal of feces from the anus, not to be confused with excretion. You do not excrete feces, you egest them. Excretion has a different definition. It is the removal of waste products of metabolism. And just to touch on a couple more definitions, ingestion is the taking in of food into the body, and metabolism is the rate at which chemical reactions take place in the body. And lastly, assimilation is building up large molecules from small molecules. Looking more closely at egestion then, so we know that's the removal of feces from the anus. Now, feces which are incredibly watery, we know is diarrhea, which is very, very unpleasant. How do we treat diarrhea? Well, there's not an awful lot we can do. We do tend to recommend oral rehydration therapy, and that contains a mixture of water and mineral salts. And this is to prevent the dehydration which is associated with diarrhea, because obviously if you're losing lots of watery feces, you're not taking in enough water, so that needs replenishing. Cholera is a disease which is awful. It is diarrhea to the extreme. Now, this is caused by bacterium, and what happens is that bacteria produces a toxin. This toxin increases the loss of chloride ions in the small intestine, which leads to a really strong osmotic effect within the gut, causing massive amounts of water loss. This causes diarrhea, dehydration, loss of salts from the blood, and I know this sounds mad, but it can even lead to death. Cholera is a major cause of death in developing countries. Let's look at transport in plants. So we're going to start by looking at two vessels, two tissues you need to know lots about. That is the xylem and the phloem. Make sure you can label these both inside the root at cross-section and inside the stem. Note that in the root, the X matches with the xylem, which is why the X in the middle of the root is xylem. That's a nice way of remembering. Whereas the circles around that X are the phloem. Inside the stem, it's slightly different. The outer layer of tissue is the phloem, the inner layer is the xylem. Try and remember this because the phloem transports sugar, and aphids are little insects which bite into that stem in order to steal some food, so they're biting in so that they can reach that phloem and take the food, so the phloem is on the outside of the stem. So looking more closely at the roles, as I've already told you, phloem transports glucose, and it transports it from the leaves where it's made in photosynthesis to other parts of the plant, so to growing regions such as flowers and the tops of stems, and to storage regions such as the roots, and that's where it's stored as starch. Xylem transports water this time and mineral ions from the roots where it's absorbed through the root hair cells up the plant to the leaves in various places. So this is an important thing for you to note. Xylem only transports water upwards, phloem transports both up and down the plant. In terms of the structures, because xylem is transporting water and mineral ions, it needs to be very strong, and notice that it's made out of dead cells which are stacked on top of each other, so there's no organelles in there obstructing the flow of water. There's also lignin which helps to reinforce those walls further. Phloem has a different structure. Just list some keywords here, don't talk too much about it. It has sieve plate tubes, and it has companion cells, and those companion cells contain lots of mitochondria so that they can release energy so that sugar can be actively transported in and out of the phloem. We're now going to look at how the mineral ions enter the plant. So I think I've already told you this, but they enter by active transport. They enter against the concentration gradient. The reason being is there's far fewer mineral ions in the soil compared with what's inside the plant is greedy. It wants as many mineral ions as possible. So because it's against the concentration gradient, from a low concentration in the soil to the high concentration in the plant, it absorbs it using active transport into that root hair cell. Looking at how water is absorbed, again through the root hair cell, this is by osmosis. So it's from an area of high water potential in the soil to an area of low water potential inside that root hair cell. Lastly, how does water move up through the plant? Well, just to give you a bit of chemistry background here, remember that water leaves the leaf at the stomata by transpiration. So as the stomata open, the first droplet leaves. But because water molecules are all attracted to each other due to the presence of hydrogen bonding, this forms a continuous column within the xylem. So it's a huge chain of water molecules. So as one leaves the stomata, another one is drawn up from below, and we call this transpiration stream. You need to know about how the rates of transpiration may be increased and decreased. So transpiration, let's remind ourselves, is the loss of water vapour from the surface of the leaf, i.e. through the stomata. So we're looking at how that water is leaving the leaf. So how might we increase the rate at which that occurs? And I always say, think about washing, drying outside. What sort of conditions would you want for your washing to make it dry nice and quickly? So first of all, we want it to be dry. And the same here, this increases transpiration rates when it is dry. Why is this? And that's because there are very few water molecules in the air surrounding the leaf. There's far more water inside the leaf, therefore water will diffuse out through the stomata very quickly when it is dry. If we look at when it's humid, so when there's lots of water in the air, it feels really yucky on your skin, there's a lot of water in the air surrounding the leaf, lots of water in the leaf. There's not a lot of difference between the water molecules inside and outside of the leaf, so diffusion is going to occur very slowly when it is humid. Next up, what's going to make our washing dry nice and quickly? High temperatures. We know this, in hot countries our beach towels dry nice and quickly. Same with transpiration. Increased temperature leads to increased transpiration. This is because the water molecules have more kinetic energy and therefore they can simply diffuse faster out of the leaf. Next up, we want wind for our washing, and again windiness will increase transpiration rate. The reason being that the wind blows those water molecules away from the surface of the leaf, effectively increasing the concentration gradient, so diffusion will occur nice and quickly. And then the last point, which has nothing to do with washing drying, is sun. So when it is sunnier, you will find that there is more transpiration. The reason being that because it's sunny, the plant wants to photosynthesize a lot. So it opens its stomata in order to let in that carbon dioxide which is needed for photosynthesis, and clearly because the stomata is now open, water can automatically leave. So what is the actual purpose of transpiration? Why is it a good thing? Because at the end of the day we're just seeing water leaving the plant, which seems like a bit of a waste. Firstly, the water flowing in the xylem helps to support the plant, it keeps it upright. And this is why when the plant doesn't have enough water, it starts to wilt, because it just doesn't have enough water to make its cells turgid. Secondly, it cools the plant. Thirdly, it provides a method for delivering mineral ions around the plant. And number 4, it supplies water for photosynthesis. Let's now look at transport in animals. So now we're going to talk about the circulatory system, so we need to look at the heart. I've already talked a little bit about multicellular versus unicellular organisms. The reason why organisms such as ourselves, humans, require a circulatory system is because our surface area to volume ratio is too small and diffusion is too slow. So we need a circulatory system which actually acts to transport oxygen around our bodies. So that's the reason why we have a circulatory system. The heart forms the epicenter of our circulatory system. It is the pump which delivers oxygen around our bodies, and you've got to know detailed information about how this actually happens. So we're going to divide the heart into 4. Do remember that we switch over the left and right sides when we're looking at a diagram. Why? Because we're picking up the heart and pushing it into our bodies. So it is the opposite way around if you actually think about it. So the 4 chambers are the left atrium, right atrium. They form the top 2 chambers. Left ventricle and right ventricle form the bottom 2 chambers. Now do remember that pulmonary means relating to the lungs. So if you have used the word pulmonary, it means that blood must be flowing either to or from the lungs. So we're going to start by picking up oxygen in the lungs. It's going to be delivered to the heart, to the left atrium, via the pulmonary vein. Remember that veins bring blood to the heart, arteries take it away. So the pulmonary vein delivers oxygenated blood to the left atrium. The left atrium contracts, forcing blood into the left ventricle. Do you remember that the valves open here, and they are the bicuspid valves? They open to allow blood to flow from the atria to the ventricles. The left ventricle contracts, forcing blood into an artery. This is the aorta. It is the main artery delivering oxygenated blood around the body. So that blood goes and delivers oxygen. The oxygen is removed and used by respiring cells. Clearly the blood will now be deoxygenated, and it needs to return to the heart so it can be pumped on further to the lungs. So it's going to return to the heart via a vein. The vein is the vena cava, and it's going to take blood into the right atrium. The right atrium contracts, forcing blood through the tricuspid valves into the right ventricles. The right ventricle contracts, forcing blood into an artery. This artery is clearly going to be flowing to the lungs so the blood can be oxygenated, and this is why it is the pulmonary artery. So that's our overview of blood flow around the body. Let's notice a couple of things about the heart. Firstly, that the walls of the ventricles are thicker than the walls of the atria. Why? Because they need to pump at a much higher pressure to deliver the blood much further. After all, they're delivering blood to both the lungs and the rest of the body. All the atria are doing is pumping to deliver the blood slightly lower, a couple of centimetres lower into the ventricles. Why is the wall of the left ventricle thicker than that of the right? Again, it's a distance thing. Blood from the left ventricle is going all around the body. Blood from the right ventricle is simply returning to the lungs. Your heart is here, your lungs are here. It's not too far. Why do we call this system a double circulatory system? Well, that's because the blood passes twice into the heart for every once it travels around the body. Simpler organisms such as fish have a single loop, so the blood just keeps passing from the gills to the heart around the body, round and round and round. They're not as efficient at oxygenating their bodies as we are. You've got to know quite a lot about the various vessels that travel around the body. Just remember that arteries carry blood away from the heart, and do remember that pulmonary means relating to the lungs, and that hepatic means relating to the liver, renal means relating to the kidneys, so renal failure, kidney failure, hepatitis, disease of the liver. So these words, if you do know what they mean, it really helps, because then when we're looking at what's the name of the vessel supplying the liver, well, it's hepatic. It's coming from the heart, which means it's an artery, so it's the hepatic artery. What is the name of the blood vessel entering the kidneys? Well, it's coming from the heart, so it's an artery. It's going to the kidneys, so it's renal, so it's the renal artery. The vessels leaving the organs, well, these have obviously got to return to the heart to become oxygenated, so they're going to be veins. So the vessel leaving the liver will be the hepatic vein. Just note the hepatic portal vein, that's just the name of the vessel which shunts blood from the digestive system to the liver, and that's the only weird one you need to know about. Looking at coronary heart disease now, the coronary arteries. Coronary means relating to the heart. So the heart has its own special network of vessels which supply the heart with its own supply of oxygen. It can't actually obtain its oxygen needs from the blood flowing through it. It has to have its own special set of vessels. We call these the coronary arteries, and they're famous because this is how people get heart attacks. They get blocked, they get obstructed, and it does mean that oxygen can't reach the heart muscle, so part of it dies, which is what a heart attack is. So first of all, what factors increase your chance of getting coronary heart disease? So that could be a sedentary lifestyle, so lack of exercise. It could be your diet, eating diets high in fat and sugar. It could be inheritance, so genes. Some people are just more susceptible than others because of genes that they've received from their parents. It could be diabetes. Diabetes and coronary heart disease are very closely linked. Stress as well. People shouldn't get too stressed because that can put a strain on their heart too. In terms of what happens in coronary heart disease, how a heart attack might occur, what you find is that fatty deposits get offloaded in the walls of the coronary arteries. This obstructs the blood, meaning that less oxygen can reach those respiring cells. Because they're not receiving enough oxygen, they have to anaerobically respire. Remember, that produces lactate, which slowly poisons the muscle cells, and then eventually there isn't enough oxygen, so the heart dies and those cells die. Let's look at how a heart rate increase is brought about during exercise, for example. So clearly when you exercise, you're going to produce more carbon dioxide. Why? Because your muscles are respiring more. So that carbon dioxide flows in the blood and it is detected by both the aorta and the carotid artery. This sends impulses or messages to the brain, particularly the medulla part of the brain, and specifically the accelerator nerve. Now that accelerator nerve causes an increase in heart rate so that more oxygen can be delivered to your muscles and so that more carbon dioxide can be removed. Looking now at the structure of arteries, veins and capillaries. So let's start with the arteries. They have a narrow lumen. Remember, the lumen is the hole in the artery. It's like the hole in a straw. So it's narrow. This clearly means that blood is going to be forced through at a high pressure. Because it's at a high pressure, it means that the walls of the arteries need to be very thick to withstand this pressure. Be nice and detailed here and state that they have thick muscle and elastic fibre walls. Looking at veins now, in veins blood travels at a much lower pressure. This is because veins have a much wider lumen. The walls therefore need to be much thinner, so they have thin muscle and elastic fibre walls. They contain valves, and these valves prevent the backflow of blood because that blood sometimes travels so slowly it's liable to start backing up. We don't want that to happen, so the valves force it to move on in the right direction. Capillaries now. Capillaries are the tiny vessels that supply all our cells with oxygen. They are 1 cell thick, and this enables a very short diffusion distance, and they have an extremely narrow lumen. We're going to start by looking at the components of blood. That will be red blood cells, white blood cells, plasma, and platelets. Now remember, plasmid is that liquid which actually acts as a suspension. It carries these various cells around the body. What sorts of things are transported in plasma? These are going to be our products from digestion, so things like glucose, amino acids, so soluble products. There'll also be hormones being transported. There'll be urea from the liver, which needs to be taken to the kidneys so it can be excreted, and carbon dioxide. Platelets. These are small fragments of cell. They clot the blood at the site of a wound, so they're very important in forming scabs. They convert soluble fibrinogen into insoluble fibrin, and that forms the meshwork which seals the wound. Looking more closely at red blood cells, we need to look at the structure of red blood cells so we can see how they're so well adapted for their function. They have a biconcave dish shape. This means they're donut shaped, and what this does is it maximizes the surface area to volume ratio, ensuring that they can transport as much oxygen as possible. The absence of a nucleus also means that there's more room for oxygen. And do mention that they contain a pigment called hemoglobin. Remember, iron is needed for hemoglobin production. I mentioned that earlier in balanced diet. And that hemoglobin binds to oxygen, forming oxyhemoglobin. Next topic, white blood cells and the immune system. So let's first of all discuss how we prevent pathogen entry in the first place. So remember, our skin acts as a barrier. Our hydrochloric acid in our stomach helps to destroy pathogens. Our tears prevent pathogens entering our eyes, and also your eyelashes. But what happens once those pathogens actually enter our body, enter our bloodstream? Clearly we can't stay ill forever and ever and ever. So there are mechanisms in place which actually act to remove those pathogens. The two mechanisms you need to know about are white blood cells, and they are the phagocytes and the lymphocytes. So starting with the phagocytes, remember that they engulf or ingest pathogens by enclosing them inside a vacuole, and then digestive enzymes are secreted which destroy the pathogen. The second type of white blood cell is the lymphocyte. The lymphocyte is far more complicated. and it works by recognising the antigen on the pathogen. It secretes lots of antibodies which destroy that specific pathogen, and in this way the pathogen is destroyed. Now, it has various modes of action which help to increase the pathogen destruction. First of all, it labels the pathogen, making it easier for the phagocyte to recognise it and therefore engulf it. It neutralises any toxins produced by the pathogen, and it also causes the bacterial cell to burst open on occasion. Lastly, it makes the pathogens stick together. With that answer relating to the lymphocytes, notice that I use lots of keywords, antigens, antibodies, for example. Try and include as many keywords as possible. Just shove them in your answer, because if you look at mark schemes, they'll be underlined as being worth a mark each, so it's worth writing them in anyway. Don't just keep repeating yourself. You need to insert lots and lots of keywords here. Lastly, vaccinations, vaccines. So obviously when you're going on holiday somewhere tropical, you might need to go to the doctor to get some vaccines. Now, what these are is they're injections containing either a dead, weakened or attenuated form of the pathogen. That means it contains the pathogen's antigens. Now, when those antigens enter your body, clearly your lymphocytes are going to be set off and they're going to produce antibodies which actually respond to those pathogens, and some of those lymphocytes turn into what's called memory cells. Those memory cells remain in your body, and therefore, if you become infected at a later date with a much larger amount of that pathogen, there are memory cells already in place which can secrete antibodies very quickly, much faster, much sooner, and in a much larger quantity. And what that means is that pathogen can be destroyed before it can take a hold of your body. So the whole point of vaccination is to inject a harmless version of that pathogen, so that if you accidentally become infected at a later date, it can be destroyed before it can take hold. So let's look more closely at some vaccinations which are made. So dead pathogens are used to immunise against whooping cough. We used a weakened form of the pathogen to treat measles and also tuberculosis. And then lastly, we just inject the antigens themselves when we're treating influenza. Moving on to the breathing system, so we are looking at our lungs and all the vessels relating to that. So we're going to start with our mouth. It leads down to our windpipe, which we're going to have to call the trachea. The trachea branches to form two bronchi. Further branching occurs, which is the bronchioles, and that ends in lots of air sacs, which we call alveoli, and they're surrounded by a network of blood capillaries. Now, if you actually think about it, it is very much like a tree, because the trachea is represented by the trunk. The big branches are the bronchi, smaller branches are the bronchioles, the alveoli are similar to the leaves, so do bear that in mind. Now, we need to talk about how those bronchi and bronchioles are kept clean. That's due to the presence of two types of cell, the goblet cell and the ciliated cell. The goblet cell, first of all, secretes mucus. That mucus is good because it traps bacteria, pathogens, and it stops them from entering your lungs. This is when the cilia come in, because remember, they have hair-like projections which waft, and they waft that mucus, which is covered in bacteria, up to your mouth, where it can be swallowed and destroyed by your stomach acid. Now, what is ventilation? Well, that's simply taking air into and out of your lungs. Let's look in great depth now at an inhalation, so a breath in. So first of all, the external intercostal muscles contract, the ribs move up and out, the diaphragm contracts, and what this means is that it flattens, and together all these processes increase the volume within your thorax. Because there's an increased volume, clearly the pressure will decrease, because the same amount of air is now present in a larger volume, and this means that air will be sucked into your lungs, causing your lungs to inflate. Now we want to take an exhalation, we want to breathe out, so the opposite takes place. This time the internal intercostal muscles contract, the ribs move down and in, the diaphragm relaxes. This has the effect of reducing the volume inside the thorax. The pressure increases relative to the pressure outside the body, and therefore air is sucked out of your lungs and your lungs deflate. A small thing on looking at the composition of gases in your lungs, clearly air that you inhale will contain more oxygen than the air you exhale. Why? Because the whole point of breathing in is to get oxygen into your lungs so it can diffuse into your blood and then be taken around your body for respiration. Respiration obviously produces carbon dioxide, which is why when you breathe out, the air contains more carbon dioxide than the air you breathed in. Looking more closely at the alveoli, because this is where gas exchange takes place, where oxygen moves into the blood, carbon dioxide leaves, we need to look at the adaptations of alveoli for gas exchange. So clearly they have a very large surface area. They are thin, which gives you a short diffusion distance for that oxygen and carbon dioxide to move across. They are moist, which helps the gases to dissolve. And I can't think of anything else. So that is the adaptations of alveoli done. The next topic we need to tackle is respiration, both aerobic and anaerobic respiration. Remember, it's the process carried out in mitochondria which releases energy. So what is this energy used for? Well, it's used in cell division, it's used to build up large molecules from small molecules, it's used in active transport, and it's used to contract our muscles. Let's look at the equation for aerobic respiration now. So you need to take oxygen into the body, you need to add that to glucose, an arrow, and then what's produced is carbon dioxide and water, and energy is released. Again, you need the balanced symbol equation here, and it's again all the sixes. I hope you've noticed that photosynthesis and respiration are the same equation, just reversed. So in terms of the balanced symbol equation, you're looking at C6H12O6 plus 6O2 arrow 6CO2 plus 6H2O plus energy in a square bracket, if you so wish. Now, aerobic respiration involves the use of oxygen, and that's the type of respiration that we carry out ordinarily. Now, sometimes we have to carry out anaerobic respiration, which tends to be when we've exercised and when we can't take enough oxygen into our bodies, and this involves the incomplete breakdown of glucose, and we find that lactate is produced instead. Now, lactate is pretty poisonous. It's what gives our muscles cramp, and we need to remove that lactate, so we have to take in more oxygen, which we call the oxygen debt, in order to break down that lactate to ensure the glucose is completely broken down. Now, there are two places where anaerobic respiration takes place that you need to know about. So I've already mentioned one, which is in your muscles. So I've given you all that information about the oxygen debt. It's when you can't take enough oxygen into your body, and that's because you've been doing something pretty strenuous like sprinting. You also need to know about anaerobic respiration in yeast. Remember that yeast is a type of fungus, and yeast anaerobically respires, and it breaks down glucose into ethanol plus carbon dioxide, and these are very useful industrial processes because remember the carbon dioxide is used to help bread rise, and that the ethanol is used in beer making. Moving on to excretion, so both in plants and humans. Obviously, when we're talking about humans, we'll be really looking at the kidney. Let's start by looking at the definition of excretion. It's the removal of waste products of metabolism from the body. Looking at excretion in humans, what sort of substances are excreted? First of all, sweat from the skin, urea from the kidneys, carbon dioxide from the lungs. Do note that feces are not excreted, they are digested, and I already touched on this in the nutrition part of this video. So remember, with the kidney we have the nephron. This is when you take the kidney and you zoom in and you see that there's a Bowman's capsule followed by the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and then lastly the collecting duct. So let's have a look first of all at what happens at the Bowman's capsule. That is a process of ultrafiltration. Ultrafiltration is when small molecules are filtered out of the blood into the Bowman's capsule. Such molecules include urea, sodium and chloride ions, glucose and water, and remember that these are forced out under pressure. So how does the structure of the Bowman's capsule and the blood vessel flowing into it, the glomerulus, actually aid this ultrafiltration? The reason is because the blood vessel entering the glomerulus is wider than the one leaving. This creates a huge amount of pressure which actually forces those small molecules out of the blood into the Bowman's capsule. Which molecules don't enter the Bowman's capsule? Well, that's proteins. Why? Because they're too large to pass through the basement membrane. Now we've entered the proximal convoluted tubule, we need to know what happens there. The process of selective reabsorption occurs here. Now this is when all glucose, some ions, and none of the urea is removed from the proximal convoluted tubule back into the blood. This is occurring against the concentration gradient, so it requires energy. Now we're moving on to the collecting duct, because we now need to look at water reabsorption, and this is all to do with osmoregulation. Now osmoregulation is the control of water content in the blood, and obviously sometimes we drink lots, so we need to wee a lot. Sometimes we drink very little, so our wee will be very, very concentrated and small in volume. And now we're going to look at the mechanisms involved in controlling the amount of urine we produce. So let's start when we've not had very much to drink, so overnight for example, on a hot day. Clearly our bloodstream will contain little water. That passes in the blood to your kidney, specifically the collecting duct, and it basically tells the collecting duct to be more permeable to water. So it adds lots of channels into the collecting duct, which enables lots of water to leave the collecting duct and be reabsorbed back into the blood. This means that there's far less water left over to create urine. So in this situation, urine is very yellow, it's concentrated and low in volume. Let's look at the opposite effect. So when we have had lots and lots to drink, it makes the walls of the collecting duct less permeable to water, meaning that more water stays inside the collecting duct and that less is available to be reabsorbed. This means that there is more water flowing from the collecting duct to the bladder, and therefore resulting urine is high in volume, not very concentrated and very pale in colour. Just to touch on the name of some key vessels, the vessel linking the collecting duct to the bladder is known as the ureter. The vessel taking urine out of the body, so from the bladder, is known as the urethra. And if they ask you in men what two substances does the urethra transport out of the body, that would be both sperm and urine. Definitely not water. Some people in the past have written water. That is really silly. That is not the answer. Looking at the general structure of a kidney, you can see the outermost layer is known as the cortex, the middle layer is known as the medulla, and then the ureter passes out, and then we've obviously got the renal artery and the renal veins supplying and removing blood. We're talking about the nervous system here. So let's start by looking at, again, what is a stimulus? It is a change in the environment. Now that change in environment is detected by various sense organs, and you need to be aware of their names and what they're actually receiving information about. Starting with the eye, that receives light energy. The ear receives sound energy and kinetic energy. The muscles in your skin receive kinetic energy. Your tongue receives chemical energy. So your nose, it also receives chemical energy. Why? Because that's chemicals in food and that's what you smell. And then lastly, your skin receives kinetic energy and heat energy. So there are two types of communication you need to be aware of: the nervous communication and hormonal communication. So hormones are chemicals which travel in your blood. They send signals and messages to various parts of your body, whereas the nervous system involves the use of electrical impulses. So let's have a quick comparison between the two. First of all, the nervous system is much faster than the hormonal system. Clearly, electrical impulses will get to their location much faster than hormones travelling in the blood. The nervous system involves very localised responses, so the electrical impulse will be locating a very specific effector muscle, whereas the hormonal system has far wider spread effects. The nervous system responses are short-lived, whereas the hormonal system involves much longer-lived responses. And the last thing, which I've already mentioned, but just to really point it out to you, the nervous system involves electrical impulses, whereas the hormonal system involves the use of chemical messengers. So we're going to take the nervous system in greater detail now. So let's first of all look at what a stimulus is. So that's the change in the environment, and obviously that causes the response that the nervous system brings about. Do be aware of what the central nervous system is. That consists of both the brain and the spinal cord. Let's go through all the steps involved in a regular nervous response, one which does not involve a reflex action. So I'm going to use picking up a book as an example. So first of all, we need to list the stimulus, which is seeing or viewing the book. This is picked up by receptors, and these receptors will be in your eyes, your photoreceptors on your retina. They'll send electrical impulses along your sensory neuron to your central nervous system. Electrical impulses then pass along your motor neuron to your effector, and this will be muscles or glands, and in this particular case it will be muscles which will contract to pick up your book. Remember, an effector is either a muscle or a gland, so a muscle responds by contracting, a gland responds by secreting hormones. Don't forget the role of the synapse. So the synapse is the gap between two neurons, and this is where a neurotransmitter is released. So the neurotransmitter diffuses across that synaptic gap, that synaptic cleft, and binds to the postsynaptic membrane. So your electrical impulses change to a chemical or a neurotransmitter and then change back to an electrical impulse at a synapse. Looking now at reflex actions, remember these are faster and they are involuntary, so they do not involve a conscious part of your brain, and it tends to be in response to something painful. So putting your hand in the oven and accidentally touching one of the shelves, which is hot, this would trigger a reflex action. So the stimulus this time would be the high temperature from the oven tray. Your receptors would be on your fingers, which would receive that information about it being too hot. Electrical impulses would then flow along your sensory neuron to your relay neuron this time, so we're not involving the conscious part of our brains. The electrical impulse passes along the motor neuron to your effector, which will be a muscle in your finger which contracts to remove your hand or your finger away from that heat source. Right, we're going to look now at the eye. You need to know lots about this. It's a very important sense organ. So I'm going to start by running through its various structures and their roles. So first of all, light enters the eye and it hits the cornea. So the cornea's role is to refract the light, to bend that light as it enters the eye. The light then has to pass through the pupil. So the pupil's role is simply to allow light into the eye. The size of the pupil, however, is controlled by the iris, and that is the colourful part of your eye. Mine's brown, other people's are green or blue. And the iris has circular and radial muscles which will actually help it control the size of the pupil. We'll go into that in slightly more detail further on. So now light has entered the eye, it now hits the lens. The lens's role is to refract that light further in order to focus it on the retina. The retina contains photoreceptors. These are cells which are sensitive to light, and they are called rods and cones. Rods are sensitive in very dim light, and cones are sensitive to colour. Then we have the optic nerve, and that converts those light signals into electrical impulses which can be carried to the brain where they can be computed. Then we have a blind spot. The blind spot is simply the place on the back of the eye where the optic nerve leaves the eye, because clearly there can't be any photoreceptors where you have the optic nerve. The fovea is a place that is very concentrated with cones, so it's very good for seeing colour. We need to look a bit more now at the lens. So I've already told you that the lens focuses light onto the retina. It does this by a process of accommodation. So accommodation is all about focusing light from different distances away so that an image can be formed on the retina. So let's look at what happens when we're looking at an object far away. So when it's far away, the light is going to be coming into your eyes fairly parallel, which means it doesn't need refracting very much. Therefore, the lens needs to be thin. In order to do this, the ciliary muscles relax and the suspensory ligaments are taut, and this means you have a very nice thin lens, so it doesn't refract the light too much. Looking at an object really close to us now, the light rays are going to be coming in at a far greater angle. Therefore, the lens needs to do far more work in order to focus light onto the retina. In order to do this, the lens needs to be fat. So in this case, our ciliary muscles contract and our suspensory ligaments slacken off, meaning that our lens is nice and fat. So that's the way in which we focus on light at different distances away. We now need to look at the pupil reflex. So the pupil changes its diameter dependent on light levels. So if there's lots of light, the pupil needs to be narrower, and that's to prevent damage to the retina, because too much light entering the eye will damage the retina. If we're in a dim or dark room, the pupil obviously needs to be nice and wide in order to allow as much light into the eye as possible. We can use this response as an example of a reflex action. So previously I told you all the steps involved in a reflex action, and we're just going to use those steps, but we're going to apply them specifically to this example. So I'm going to use an example which is that we have walked into a very bright room. So the stimulus will be lots of light. The receptor will be the rods and cones on your retina. Then the sensory neuron will pass along the optic nerve to the brain, and there will be a relay neuron. Then we have a motor neuron, which is also passing along the optic nerve, and it will end in an effector. And in this situation, the effector is the muscles which you find in your iris, so your circular and radial muscles. And you'll find that your circular muscles will contract and your radial muscles will relax, and that will act to narrow or constrict the pupil. Now looking at a separate hormone, which is adrenaline. Remember, that is the fight or flight hormone. It's released in bucket loads when we're under conditions of stress. And it's good to imagine a really angry cat when you're thinking about the effect that adrenaline has on your body, because if you imagine the cat, then it will actually help you remember every single thing that happens to you when you have adrenaline coursing through your body. So this cat's hairs have stood on end, and that's because it wants to appear scarier to its opponent. Its pupils have dilated, and that's to allow more light into its eyes so it can see more clearly. Its heart rate, not that you can see this, but adrenaline has caused its heart rate and breathing rate to increase. Why? The heart rate increases to deliver blood faster around your body. Your breathing rate increases to allow more oxygen into your body for respiration. Blood is diverted from your digestive system to your active muscles in your arms and your legs, and that's so you can either fight or run away if needs be. So yeah, try and remember the cat, at least for the hair standing up on end and the pupils dilating. Another thing to do with homeostasis, we're looking at blood sugar. So how do we decrease our blood sugar levels after we've eaten? Well, we release a hormone called insulin. Now, insulin is secreted from the pancreas, and it causes glucose to be converted from its soluble form into glycogen, which remember is insoluble, and that can be stored in the liver. Let's look at the role of the skin now. So first of all, the skin acts as a barrier, and that actually helps prevent pathogen entry, because we often have viruses and bacteria landing on us, but because the skin is such an amazing intact organ, it does actually prevent those microorganisms entering our body. It's also waterproof, which is why you don't swell up when you go swimming. It forms a very tough outer layer just generally to prevent you from physical harm if you knock into something. It's a sense organ for pressure, touch and pain. And lastly, it controls our heat loss. How? By either sweating or the hair stand up in order to trap some insulating air close to our bodies. Let's now look at what happens when we're too hot and too cold. So we're looking further at the homeostasis topic, which remember is all about controlling a steady internal environment within our bodies. So let's start by looking at when we are too hot. Well, first of all, an uncomfortable thing happens, which is that we sweat. The reason being that when sweat evaporates, it acts to cool the body. Vasodilation occurs, and that's the arterioles in our face. They dilate, they become wider. What that means is that the blood flows closer to our skin, and therefore heat can be radiated more easily. And also when we're too hot, the hairs lie down on our body, on our arms, and that's just so that less insulating air is trapped close to our skin, because remember, air is a good insulator, it keeps you warm. So by making our hairs lower, it means we're kept less warm. So if we're too cold, clearly our hairs will stand up on end, so that more insulating air is trapped close to our skin, and that actually acts to keep us warm. We shiver, the reason being that that muscle contraction releases heat energy. And lastly, the opposite of vasodilation occurs, vasoconstriction. The arterioles in our face constrict, they narrow, bringing blood away from the surface of our skin, so less heat is radiated.
Let's look at how blood sugar levels are controlled, and we need to know lots of hormones and the effect that they have on blood sugar. We're going to take two scenarios. So first of all, after a meal, clearly your blood glucose levels will increase because you've just eaten. It's important that we reduce those blood glucose levels. So the pancreas, remember which organ does this, so it's the pancreas, releases the hormone insulin. And that insulin travels to the liver and causes it to convert soluble glucose into an insoluble compound known as glycogen. And as you can see, blood glucose levels will therefore have decreased. After exercise, or when you haven't eaten for a period of time, clearly your blood sugar levels will have decreased. So glucose levels have decreased. The pancreas again is involved and it releases a hormone, this time called glucagon. And please notice the spelling of glucagon and glycogen. They're incredibly similar sounding words, and it's so essential you spell them correctly, because I know there'll be a point in the mark scheme being like reject if they've spelt it wrong. So make sure you take pains to learn that spelling correctly. This time the glucagon travels to the liver and it causes the insoluble glycogen to be converted back into soluble glucose, which then gets released into the blood, and subsequently you see an increase in blood glucose levels, which is all good. Now, famously, a disease when you don't control your blood sugar levels appropriately is called diabetes, and there's type 1 and type 2. Type 1 tends to be the one you get as a child, and we call it early onset for this reason, and it is inherited, so you get it through your genes, so you inherit it from your parents. And here you'll have to do insulin injections because effectively your pancreas isn't producing enough insulin. In terms of symptoms, so how you might know that you have diabetes, first of all, you'll get excessive thirst, so you'll want to drink all the time. As a consequence of that, you'll produce lots of urine because you've taken on so much water and liquid. You get increased hunger, sweet smelling breath, and the biggest giveaway is if the doctor tests your urine and there's glucose in the urine, that can be an indicator of diabetes, because really we know that the glucose should have been reabsorbed in the proximal convoluted tubule of the kidney, so glucose in the urine would indicate that there's something wrong.
So we're going to take plants first of all, and we're going to look at tropisms. First of all, what is a stimulus? And that is a change in the environment. So what sort of stimuluses do plants need to respond to? Clearly they need to respond to the amount of light, and they actually also respond to gravity. So we call a plant's response to light a phototropism. A plant's response to gravity is known as geotropism. So how do organs of a plant react to various tropisms? So let's take phototropism first of all. Clearly a stem shows positive phototropism. Why? Because it grows towards the light. Roots show negative phototropism because they grow away from the light. Let's look at gravity now. Roots obviously show a positive geotropic response because they grow down in the direction of gravity, whereas stems show a negative geotropism because they grow away from gravity. Looking more closely now at how these changes are brought about to the stem and the roots, we need to understand the role of auxins. So what are auxins? Well, they are plant hormones. Let's now explain how a plant stem may bend towards the light, towards the sun, because you do see particularly sunflowers bend towards the sun. That's because the auxins concentrate on the side furthest from the light source. This causes cell elongation on that side, so the cells get longer, and therefore the plant stem bends towards the light. They do like to show you various experiments, and an important part of this will be the coleoptile, which are these really boring little seedlings. Now note that they are cereal seedlings, and they are simple plants used to investigate tropisms. So do be aware of how they respond if you chop their tops off, if you put a mica sheet in between the top of the stem and the bottom of the stem, if you use agar jelly. You do need to know different responses. Lastly, the role of a clinostat. This is another boring piece of equipment. So a clinostat is a device used to remove any stimulus. So for example, it negates the effect of gravity or negate the effect of sunlight.
Drugs, a scary topic because if drugs are misused, they can cause lots of issues. But the one thing to notice is that drugs aren't all bad. I think people tend to think of heroin, cocaine, ecstasy, etc. when we talk about drugs. Yes, indeed, these are drugs, but remember there are plenty of therapeutic drugs, things like paracetamol, aspirin, morphine, and these have all been proven to really help human health, to alleviate symptoms, produce pain relief. So it's not that all drugs are bad. It's just that you need to remember that a drug is actually just a chemical which affects the body. And there's so many different categories of drug. One very important category includes the antibiotics. Crucially, these are given to you if you have a bacterial infection, so they're used to kill bacteria. And they do this by damaging the bacterial cell wall, meaning that it bursts and can no longer replicate. They do not work on viruses. This is commonly misunderstood. People go into the doctors with a cold and expect antibiotics, but unfortunately for them, these colds are caused by a virus. Its name is rhinovirus. And if you ask for antibiotics, the doctor simply won't give them to you because it will not have any effect whatsoever. Increasingly though, we're seeing antibiotic resistant bacteria, which is incredibly alarming. So antibiotics are being given to treat these bacterial disease where historically it would have killed all the bacteria, but you're finding the bacteria are surviving and the antibiotics are having no effect. And this is all due to natural selection because some of those bacteria mutate, and some of these mutations mean that the bacteria is no longer affected by the antibiotic. Clearly these bacteria will survive. They'll reproduce, and before long you end up with an entire colony of bacteria which is resistant to an antibiotic, which is becoming a real concern. It's incredibly scary. So how can we reduce the chance of antibiotic resistance? First of all, complete your course. And what that means is if you're prescribed antibiotics, you must finish the course. You mustn't just stop taking them the moment you start feeling better. So if the course is 3 weeks long and you started feeling better after 2 days, it doesn't matter, you need to complete the course. And secondly, you should only be prescribing antibiotics when essential. So historically, pigs and things used to be given antibiotics even when they weren't sick, just to stop them getting sick in the first place. And this was responsible for a lot of the resistant strains that we see today. So you need to know that antibiotics don't work on viruses. And that's not just because they don't have a cell wall in the way that bacterial cells do, but it's also because viruses work in a very different way to bacteria. So they actually infect their hosts, they get inside the cell, and so they're incredibly hard to spot by either the immune system and the antibiotic. We need to touch in more detail on two very famous drugs, alcohol and heroin. These are both incredibly addictive. Alcohol seems to have less of a bad name attached. I think it's because it's so readily available. You can buy it in any supermarket and it's more socially accepted, whereas heroin, people tend to inject it. It's got quite a dark stigma attached to it, but the point is they're both exceptionally dangerous. Now, what effect do they actually have on the body? Well, they're bizarrely depressants. I know people take alcohol because they think it makes them feel good and it makes them feel less inhibited, so they're more sociable, etc., etc. But they actually act as depressants, and that means that they slow your reaction time, which is why you really should never drive under the influence of either alcohol or heroin, because you're less likely to spot hazards, you're less likely to put the brakes on, and you're more likely to kill other people. So never, ever, ever drive under the influence of either of these things, and as we know, it is completely illegal. You see reduced self-control with the use of both, which again is why people like to take them. But if that goes too far out of control, you see a negative social impact. So people don't like it. People don't like drunk people. They're scared of them. You never know what they're going to do next. And with addiction comes withdrawal symptoms. So if you've become addicted and you're used to taking these drugs all day long, every day, and all of a sudden you're like, no, this is having no good effects on me, I need to stop taking them. You end up with nasty withdrawal symptoms, things like diarrhea, hope I've spelled that right, vomiting, fevers, etc. They're pretty horrible drugs to come off. And then if you do carry on taking them, you're obviously susceptible to poor health. So with alcohol, you see liver disease or liver cancer, and we call a disease liver cirrhosis. Heroin you have problems, which I mentioned when I was talking about AIDS and HIV. You have people sharing needles. And if you're sharing a needle with someone that has HIV and you get their infected blood into your blood, then you're going to get HIV on top of all your other problems.
Smoking, which is one of my favourite topics, I think this is really interesting. I'm going to give you a very detailed answer as to how smoking affects your body. I'm going to take the various components in turn and talk about the effect that that has on your body. So let's start by looking at why people smoke. It's because it contains the drug nicotine. Nicotine is addictive, it's why people really struggle to give up. It also has adverse effects on the body by making your blood more viscous. That means it's stickier, it's thicker, and is more liable to have high blood pressure and cause blood clots. Looking at carbon monoxide now, you must have heard of carbon monoxide poisoning on TV adverts. It's a very dangerous gas, it's toxic, and that's because it combines irreversibly with the hemoglobin in your red blood cells. It forms carboxyhemoglobin, and that prevents oxygen transport and effectively suffocates you. We also have tar inside cigarette smoke. Now that acts as a carcinogen, which means it's a cancer-causing agent, so it contributes to lung cancer. It also coats your cilia, and by coating your cilia, it prevents them from wafting, it paralyzes them, and prevents that mucus, which is laden with bacteria, from leaving your lungs, leaving your airways. And because of that, you're more susceptible to infections such as bronchitis, and that's where people get a really characteristic throaty cough, because the removal of this mucus isn't happening passively anymore. They're actually actively having to cough it out, which is why they get that pretty unpleasant cough. Emphysema is a pretty nasty disease relating to smoking. What happens here is the alveolar walls are damaged, they break down. This decreases the surface area available for gas exchange and effectively means that people can't get enough oxygen into their lungs and they get very short of breath. We are now moving on to reproduction, so we'll start by looking at the difference between sexual and asexual reproduction. Hopefully sexual reproduction is nice and straightforward. Clearly you need two parents, and it involves gametes. Remember, the gametes are eggs and sperm because these are sex cells. So the sperm and egg meet at fertilization. The first cell formed is known as a zygote, and then it divides by mitosis to form your multicellular organism. And clearly, due to the production of gametes, these offspring will be genetically varied, which is a really important thing to note with sexual reproduction, is that it produces genetically varied offspring. So if the environment changes, it means that some individuals will be better adapted whilst others will be less well adapted, and that's when natural selection comes in, but we'll come across that later on in this video. With asexual reproduction, you only need one parent. It's a much quicker process. You end up with genetically identical offspring, and this is why it's good when conditions are unchanging. It's a great way of quickly producing large numbers of identical offspring, which we call clones. There is no gametes, no fertilization, and no zygote is formed. Some key definitions now. So fertilization, that's obviously the joining of an egg and a sperm. A zygote is formed, and remember, it undergoes mitotic cell division, so lots of mitosis takes place to form 2 cells, 4 cells, 8 cells, 16 cells. So if they ask you how a 32 cell embryo is formed, sperm and egg, gametes, make sure you include lots of keywords, join at fertilization. The zygote is formed, it divides by mitosis to form 2 cells, 4 cells, 8 cells, 16 cells, 32 cells. And that's how you get lots and lots of marks by not writing very much, by including keywords such as gametes, mitosis, etc. So in order to look at reproduction in plants, we obviously need to look at the structure of a flower. You need to know the names of the different parts of the flower, and know that the flower is separated into both male and female parts. So the male part consists of the anther and the filament. So the filament is actually the thing that supports the anther, whereas the anther contains the pollen grains, and they're essential because they're the male gamete in a plant. The stigma and the style and the ovary are the female parts of the plant. The collective noun for them is the carpel, and everything else is just extra detail that will actually help attract an insect or will help with the wind pollination aspect of sexual reproduction in plants. So now we know about the structure of the flower. That will help us understand what pollination is, fertilization, etc., etc. So we're now going to look at seed formation. It's important that you know every step involved in this because you could be asked a 5-marker on this. So first of all, pollen from a male anther will land on the female stigma. A pollen tube will grow down the style and digestive enzymes will help break down the wall of the ovary. At this point, the pollen will meet the ovule, which is the female gamete, and fertilization takes place. The ovule then goes to form the seed. The ovule wall forms the seed coat, and lastly the ovary wall will form the fruit. So make sure you list those last steps involved. But first of all, let's understand what we mean by the term pollination. You've got to be super specific here. So remember that the male gamete in a plant is pollen, the female gamete is the egg, but all pollination is transferring that pollen to a separate plant. So your perfect definition here is that pollen produced on the anther, which is a male part of the plant, is transferred to the stigma, which is the female part of the plant, on a second plant, and the perfect definition will pop up now so you can see exactly what you need to write. Self-pollination, obviously as the name suggests, that's when the plant does it itself, so that's where pollen from a single plant will land on the stigma of the same plant. Fertilization now, so remember fertilization in humans is all to do with sperms and eggs fusing. This time, as we're talking about plants, it's about the pollen fusing with the egg. Looking now at the difference between insect and wind pollinated plants, so when we're talking about the insect pollinated one, think about all the ways in which these flowers, these plants make themselves appealing to insects. So first of all, they have bright, large, colourful petals. These are flag-like structures and they literally draw the insect's attention. So they're like, right, come and pollinate me. Secondly, they have a nectary. The nectary is essential because that's actually why the insect is visiting the plant in the first place. It's not doing the plant a favour, it doesn't give her a damn about the plant. It's simply trying to obtain the sugar from that nectary, which is why insect pollinated plants have a nectary. They have enclosed stigma and anther, and what that means is that the insect is forced to rub against the pollen or the stigma when they enter the plant to find the nectary, so they're more likely to pick up the pollen. They have a strong scent so that the insect can smell them, and that's everything I can think of. Looking now at a wind pollinated plant, it's going to be very different from an insect pollinated plant, largely because it's the wind blowing that will actually blow that pollen away to a separate plant, which is why it makes sense therefore that the anther are very exposed, so that when the wind blows, the pollen will literally blow off the plant and be carried by the wind elsewhere. They tend to be dull coloured, have small petals, no scent, for the same reason, because the wind can't see these things because it's wind, so there's no need to have flag-like petals. There'll be an absence of a nectary, again because the wind doesn't need sugar. They have small petals, small pollen grains, feathery stigma. Just list these all out and you'll be fine. Next up, we need to look at germination. So first of all, what is germination? This is obviously what happens when you plant a seed and it effectively pops out and starts growing, but you need a detailed answer. So first of all, the seed coat bursts. The radicle is the name for the small root that appears, and that starts to grow downwards. A small shoot will appear, and obviously that starts to grow upwards. And the seed's food store is used up because the plant can't photosynthesize until it grows its first leaves. In terms of the conditions needed for photosynthesis, remember the mnemonic WOW, standing for warmth, oxygen and water. So all these things are needed in order to enable a seed to germinate. Moving on to the male and female reproductive system in humans now, and there's a new emphasis for you guys on the various roles of different components of the reproductive systems. So let's start with the female reproductive system. So starting from the beginning, you've got the vagina, the entrance to the uterus is the cervix, and then branching off the uterus you've got the fallopian tubes or the oviducts, lastly ending in the ovaries. So what is the role of the ovaries? Well, it's to manufacture eggs. The role of the fallopian tube is to deliver eggs to its entrance, so that's where fertilization takes place. The uterus is obviously where the zygote embeds. It develops into an embryo, into a growing fetus, and it supports the embryo through the placenta. The cervix is simply the entrance to the uterus. and the vagina is a passage leading out of the woman, and it's where the penis inserts to deposit semen during sexual intercourse. And lastly, the urethra. It's not really to do with the female reproductive system, but do remember it's a separate tube and it transports urine out of the body, so it links to the bladder. Please don't think that a woman urinates out of her vagina. Lots of people seem to be very confused on this. No, there are two separate passageways. Moving on to the male reproductive system now. So we're going to start by looking at the testes or the testicles. These manufacture sperm and also the hormone testosterone. They link to the urethra via the sperm duct. The sperm duct is simply a tube which transports semen from the testes to the urethra. The urethra is a tube which links the sperm duct to the outside of the body, and in men it transports both semen and also urine. And then lastly, there are some glands such as seminal vesicles. Some of you may not need to know this. The prostate gland, and these just contribute fluid to the semen so that it's not just made up of sperm. Lastly, the penis, it passes urine out of the body and deposits semen inside a woman's vagina. Now let's be sensible about this topic and no being silly. Now we're describing the passage of sperm in the female. So clearly I just said the sperm is deposited in the vagina. It swims through the cervix into the uterus, and lastly it swims all the way to the entrance of the oviduct or the fallopian tube where fertilization actually takes place. So the sperm has a very long way to swim. The role of the placenta, remember this is a huge organ which actually supports the growing fetus, so it provides the fetus with oxygen, digested nutrients such as glucose and amino acids to help it to grow, and it also removes waste products such as urea. Later on in pregnancy it takes over the role of producing the hormone progesterone. And progesterone is a good place to link because we now need to look at the various hormones involved in the female reproductive system. So we're going to start with FSH. Now FSH is produced in the pituitary gland, and it stands for follicle stimulating hormone, and that's a really good way of actually helping you know what it does. So its role is to stimulate the follicle is the egg, so its role is to mature the egg in the ovary. LH is also made in the pituitary gland. It is luteinizing hormone, and its role is to cause ovulation. Now ovulation is the release of an egg from an ovary. Next up, estrogen. Estrogen is produced by the ovaries. It's responsible for secondary sexual characteristics in females. So those are all the changes that occur during puberty, such as breast development, hips widening, pubic hair, armpit hair, all those sorts of things. Sexual drive develops. Its other role is to inhibit FSH, so it actually slows the production of FSH, which makes sense really, because if you've already had an egg released, you don't want to be maturing any more eggs because presumably you're going to be fertilizing that first egg. And its last and very important role is to repair the uterus lining, so it causes it to thicken in preparation for a fertilized egg. Progesterone now. Progesterone is produced by firstly the corpus luteum. The corpus luteum is just the leftover husk, effectively, when the egg is ovulated. It's the leftover structure that produces progesterone. Later on in pregnancy, the placenta, as I've already told you, takes over the role, and its role is to maintain the thickness of that uterus lining. Without a thick lining, a woman will miscarry because it really needs to be thick in order to support the growing fetus. And then, because we need to mention men, testosterone, its role is to support the development of secondary sexual characteristics. So again, the puberty changes. This includes pubic hair, armpit hair, widening shoulders, bigger muscles, voice breaking or deepening, sexual drive develops, sperm production occurs.
Looking a bit more at pregnancy, so what a lady should do to avoid causing any ill effects to her growing baby. First of all, she shouldn't smoke. Why is that? Well, because we know that smoke contains carbon monoxide, which combines irreversibly with the hemoglobin in red blood cells. And this decreases the amount of oxygen that can be carried. And you actually find that women who smoke end up with babies with a low birth mass. or birth weight. Similarly, they shouldn't be drinking alcohol. And women that drink too much alcohol, their babies are likely to suffer from something called fetal alcohol syndrome. And these babies are actually born addicted to alcohol, which is pretty unpleasant, really, if you think about it. And they show slowed development. So once you've had your baby, you need to make a decision about how you're going to feed them, and there are two main methods, so breastfeeding and bottle feeding formula, and you need to be aware of the various advantages and disadvantages. Clearly, breastfeeding, which has been used for millions of years, is highly suitable because it contains the ideal nutrient content, so the correct combination of nutrients in the correct amounts. You've got little issue with pathogens or bacteria because it's sterile. And it's low cost because obviously it's free, the mother makes it, and there's no preparation involved, so you don't need to sterilise bottles or anything because obviously the baby just drinks from the breast. Lastly, it's thought to encourage bonding, so the baby feeding actively encourages bonding between the mother and the child. However, disadvantages include the fact you have no idea how much the baby is drinking, because obviously there isn't a milliliter attached to this. You have no idea how much they've drunk. And the other problem is other people, such as the father, can't feed. Unless you express the milk and put it into bottles, you can't feed the baby. The mother's responsible solely for feeding the baby. Whereas bottle feeding formula, you can use your common sense here. What will the issues be here? Well, it's very expensive. It's not so easily digested, so you find that lots of babies that are bottle fed tend to vomit quite a lot. And lastly, it's so important that you sterilise all the bottles. If you don't, you can pass pathogens to the baby, which can obviously make them ill. However, there are advantages, whereas with breastfeeding you can't measure intake. Clearly you can measure intake with bottle feeding. You can see how much they've drunk, if it's 100 mils or 200 mils, and others can feed because anyone can make up the formula. We now need to look at methods of birth control. So this is a way of avoiding getting pregnant. They can be split broadly into 4 categories, which is natural, chemical, barrier and surgical methods, and they all vary in terms of how effective they are. Starting with natural methods, the first obvious thing you can do is abstain. So this is called abstinence, and this is basically when you don't have sex, because clearly if you don't have sex you can't get pregnant. You can also monitor your body temperature and also your cervical mucus levels, because these vary over the menstrual cycle, and at times you'll be more likely to get pregnant and less likely to get pregnant. Although it's worth noticing here that these aren't the most reliable means of controlling whether you get pregnant or not, because obviously body temperatures tend to fluctuate regardless of menstrual cycle, and the same with cervical mucus levels. Obviously abstinence, if you don't have sex you can't get pregnant. So moving on to chemical methods, you've got IUDs, and this stands for intrauterine device. So this is a device placed within the uterus which prevents the zygote from implanting in the wall of the uterus, and it could be a foreign object, and often they're called coils, which is, I think it's made out of metal, which is a little metal object placed inside the uterus. The fact that there's a foreign object means that that zygote won't implant. Similarly, intrauterine systems, they work in a very similar way. Next up we have the contraceptive pill, the injection, and the implant, and these are all really similar. So the pill is something that most people have to take every single day. It contains a combination of progesterone and estrogen, and by maintaining those hormone levels it means you can't get pregnant. An injection is just something which is given once every 3 months, which contains the hormones in a specified amount. And an implant is a small device placed under the skin in the upper arm, which releases hormones slowly over time, and all these methods will prevent you getting pregnant. The barrier methods are probably historically the ones which have been used the longest. So we're looking here at condoms, and as the name suggests, it's a barrier to the sperm, so it stops the sperm reaching the egg. So condom is obviously a plastic sheath worn over the penis that stops the sperm being ejaculated into the vagina. A femidom works in a similar way to a condom but is found inside the woman's vagina. And lastly, a diaphragm is a small plastic cup that sits over the cervix, so the entrance of the uterus, and again acts as a barrier preventing sperm reaching the egg. And then you can go very hardcore with surgical methods, so this could include a vasectomy, which is when the sperm ducts in a man are cut and tied, so obviously there's no longer a tube allowing the sperm to pass from the testes to the urethra, and therefore they can't possibly leave the body. And female sterilisation, which works in a similar way but will involve the female's oviducts rather than the sperm duct. To go into a bit more detail about the hormones found within the contraceptive pill, sometimes you find that there are progesterone-only pills, and all these do is they prevent the zygote implanting. Whereas progesterone and estrogen combined pills prevent ovulation, so they actually stop the egg being released in the first place, and therefore tend to be a better way of preventing pregnancy. Now we're looking at the opposite, so looking at becoming pregnant, and if we're not going to do normal sex, we can look at alternative methods such as artificial insemination, and this is all to do with sperm donors. So a man may decide to donate his sperm, and that gets stored in what's known as a sperm bank. I think the sperm are actually frozen, and then when the time comes, when a woman is close to ovulating, so when she's at her most fertile, then that sperm can be inserted into her vagina and she can become pregnant in the normal way. And remember that sperm can live for up to 3 to 4 days within the woman. IVF now that's quite famous because it's a method used to help couples who are struggling to conceive, and that's actually known as in vitro fertilization. And what happens here is the ovum or the egg is fertilized outside the body. Tends to happen inside a laboratory, and then once that egg has been fertilized, it's placed within the uterus of the female. And that's been an amazing way of helping couples who previously couldn't have a baby conceive. Now a fairly unpleasant topic, which is to do with STIs. So these are sexually transmitted infections. And as the name suggests, it's an infection transmitted by bodily fluids through sex. Good examples here include HIV. Herpes is another example. You could also have chlamydia or gonorrhea. I have no clue how to spell those, so I'm not going to write them here. This is becoming an increasing problem, so STIs being transmitted by people, and it's something which needs to be prevented, and there's lots of ways in which STIs can be prevented whilst people can still have sex. So that includes obviously wearing condoms, so that barrier method not only prevents pregnancy but also prevents STIs. There's testing, so that's a way of just checking to see if you have an STI so you don't continue to have unprotected sex and carry on spreading it. It's also good to have some knowledge of partner's sexual history so you can work out the likelihood of them having an STI. I realise this would be a very awkward conversation to have, but it's all about knowledge and information. The more you know, the more you can protect yourself. And things like worldwide education programs. So HIV is very prevalent in Africa, and a large part of the problem with stopping its spread is the fact that people don't understand that it is spread through unprotected sex. So it's all about educating people so they know what's happening. And then lastly, you can provide things like antibiotics, vaccinations, and antiviral drugs just to help treat the STI. So going into more detail about HIV, remember that stands for Human Immunodeficiency Virus, which later can become AIDS, which is an incredibly serious disease. Now, I've already told you it's spread by unprotected sex. Obviously, that needs to be with an infected person. And sadly, it can actually be spread from mother to child across the placenta or through milk, so feeding their child. And then syringes, so drug users are at risk of getting HIV, because obviously if they share syringes, which is a very disgusting practice anyway, but if you share a syringe with someone with HIV, their contaminated blood will obviously be injected into you, and so that can cause the spread of HIV that way. So really, really unpleasant. So what does HIV do? Well, I've already told you it can lead to AIDS. Now, AIDS is so scary because it damages the immune system. And remember, the immune system is all to do with fighting pathogens, so fighting bacteria and viruses. And what it does is it actually reduces your white blood cell count, and specifically it reduces your lymphocyte number. And you need to remember that lymphocytes are responsible for producing antibodies. So when you're infected with HIV, the number of antibodies you can produce seriously decreases.
We need to start by looking at some key definitions such as genome, and that is the entire DNA belonging to an organism. So we're focusing on the nucleus of a cell. Remember that the role of the nucleus is to control the activities of the cell, and it does this because it contains lots of genetic material. So within the nucleus, we know that there are chromosomes. There are 46 chromosomes, which is a diploid number, because remember they're arranged as 23 pairs. Remember, half the number of chromosomes is known as a haploid number. However, I digress. I don't really want to talk about that now. So the chromosomes are made up of DNA. You need to know the definition of a gene. A gene is a section of DNA which codes for a particular protein. Looking more closely at DNA now, so we need to first of all know what the structure of DNA is. Remember, it looks like a ladder. We call this a double helix, which winds itself up. It's made up of a sugar phosphate backbone, and remember that the sugar here is deoxyribose. Why? Because DNA stands for deoxyribose nucleic acid, so the sugar here is deoxyribose. There's a phosphate and sugar backbone, and then linking the two backbones, the rungs of the ladder, are bases. And remember the names of these bases: adenine, thymine, guanine and cytosine. Looking more in depth at the various names involved, nucleotide. A nucleotide is simply just 3 units made up of a deoxyribose sugar, a phosphate and lastly a base. So it could be either one of those bases I've just mentioned, so adenine, thymine, cytosine or guanine.
Sorry, is that how you pronounce it, guanine?
How do you say
I wanted it guanine.
I got taught guanine.
Really?
But it sounds weird now you're saying it. Martin's telling me that I'm pronouncing guanine wrong. My biology teacher said guanine, and I don't know how to say it any other way. Apparently he says guanine. So, you're like, what an idiot. So we need to now look at complementary base pairing. That's when the various bases pair up in a very specific way. Try and remember the shape of the letters match. So guanine or guanine always pairs with cytosine, because they're both curly in terms of the G and the C. Adenine and thymine always pair up, and that's because they're the straight letters. So use that to help you remember. Looking at the difference between RNA and DNA, the main difference here is the sugar involved. So RNA has ribose sugar as opposed to DNA's deoxyribose. And lastly, there's a change in base. There's no thymine in RNA. You've got to learn that this time it's uracil. So adenine pairs with uracil in RNA, and cytosine and guanine still pair up. RNA is single-stranded compared with DNA's double strand, so we don't see a ladder with RNA, we just see one side of the ladder effectively. Let's move on to looking what a codon and anticodon is. So a codon is just a fancy way of describing 3 bases which are found on the mRNA molecule, and they correspond to a single amino acid. So remember, protein synthesis is all about making new proteins. It's about sorting out the arrangement, the sequence of amino acids so they can actually produce a particular protein. And we're going to talk about how those amino acids line themselves up in the correct order, aka protein synthesis. mRNA are messenger RNA because it carries the message from the DNA and now needs to leave the nucleus, so it leaves via a nucleopore and attaches itself to a ribosome, which we find within the cytoplasm. Hence, the summary definition of a ribosome's role is that it carries out protein synthesis. So the mRNA attaches to a ribosome, and this is where translation begins. So there are tRNA molecules within the cytoplasm, and they have complementary anticodons. So they'll have the 3 bases which complementary pair up with the mRNA exposed bases, and at the other end of that tRNA molecule will be an amino acid, and effectively the tRNA brings that amino acid to the mRNA, the codon will match up with the anticodon, and we have our first amino acid in place. Then the next 3 bases on the mRNA will be read, and a different tRNA molecule will bring probably a different amino acid along, because if there are different bases, it will correspond to a different amino acid. So the second amino acid has been brought, and they attach to each other using a peptide bond. So that is the start of our protein chain. It keeps going, and eventually a stop codon will be reached, and that's just 3 specific bases which correspond to no amino acid whatsoever, and that will signal the end of that growing protein chain. Don't forget that haploid means containing 1 set of chromosomes, so in humans that is 23. Diploid means containing 2 sets of chromosomes, in humans that is 46. Let's look at mitosis and meiosis now. So remember, they're both types of cell division, but they're used for making very different things. So meiosis is used to make gametes, so that means it's used to make sperms and egg. Mitosis is a completely different type of cell division. You need to learn that it's used in cloning, asexual reproduction, and the growth and repair of cells. So for example, if you damage yourself, you cut yourself, it will be mitotic cell division, which replaces those cells. If you're carrying out asexual reproduction, so something like a strawberry runner producing baby strawberry plants, that will involve mitosis. The reason being is that it creates genetically identical offspring. I don't think you really need to know this, but some teachers like to just chat slightly about the different stages involved in mitosis and meiosis. I'm really only going to give you their names, and the first stage is prophase, the second stage is metaphase, then anaphase, and finally telophase. But this is a revision video and I'm not willing to talk about it anymore at this point because I don't think it's necessary for lots of you. So let's look at the differences between mitosis and meiosis. I always do this as a table because it allows me to make a direct comparison. So look at the number of cell divisions first of all. That will be 1 cell division in mitosis, 2 cell divisions in meiosis. The number of daughter cells now, so that's the number of cells produced once this cell division has taken place. In mitosis, you're looking at 2 daughter cells. In meiosis, you're looking at 4 daughter cells. I've already touched on the sorts of cells that are produced, but just to recap, mitosis produces genetically identical daughter cells. Meiosis produces genetically varied daughter cells, which makes sense. If we're using mitosis in cloning, that's genetically identical. If we're using meiosis in making gametes, it makes sense that we want our sperms and egg to all be different to each other. And do notice that the gametes will contain a haploid number of chromosomes, whereas the daughter cells produced by mitosis will contain a diploid number. So differentiation is the process whereby cells become specialised, for example nerve cells. Now a stem cell is a cell that has the potential to divide many times without being differentiated. We're going to talk about genetics now, so we need to know the definitions of lots of very key important terms, and then I'm going to show you how to do Punnett squares and pedigree analysis. So let's start by looking at what a gene is. A gene is a section of DNA which codes for a particular protein. Now there are different genes which control different traits, so for example eye colour. Now different forms of the same gene we call alleles, so you must learn that definition. So if we take eye colour for example, different alleles for eye colour could be blue or brown. You must now know the meaning of the word genotype. The genotype is the alleles an organism has. So for example, when we're talking about blue eyes, it's 2 small b's. If we're talking about brown eyes, it could be big B, little b. So when you're asked for genotype, you must provide letters. The phenotype is different. This is the physical appearance of a particular trait. So if you're asked for the phenotype of this eye colour, the answer here is blue. So the genotype would be little b, the phenotype would be blue eyes. So be very aware of that distinction. Next up, we need to know the meanings of homozygous and heterozygous. Homo means same, so it means having 2 of the same alleles, whether that's 2 big B's or 2 little b's, it doesn't matter as long as they have the same case. So they both need to be upper or they both need to be lowercase, and that is the meaning of the word homozygous. Heterozygous means different, so that means containing different alleles. So in the case of eye colour, that would be a big B and a little b. Now, a dominant trait requires simply the presence of one allele for it to exhibit itself in an individual. So brown is an example of a dominant trait because you can have two big B's or a big B and a little b and the trait will still appear. Recessive requires the absence of the dominant allele, so a recessive trait could be blue eyes because you need two little b's, for example.
A blue-eyed mother and heterozygous brown-eyed father decide to try for children. What is the probability that the children will have blue eyes? So let's work out what we have here first of all. Blue-eyed, remember blue is a recessive trait, which means that her genotype must be small b, small b. We've been told that the father is brown-eyed, which means he could be big B, small b, or big B, but the fact that he is heterozygous tells us that he must be big B, small b, and not big B. So I'll show you how to lay out your answer. This is the method you should always use. So start by writing mother and father at the top. And we know how much I love tables. So you're going to write in your table phenotype, genotype, and lastly gametes. Remember these are sperm and eggs. So the phenotype, this is the physical appearance. We can see from the description that the mother has blue eyes and the father has brown eyes. The genotype, so these are the alleles that each parent has. I've already written these out, so it's small b, big B, little b. The gametes, just split these up because this is saying what the eggs and sperm will be. So just write out what you wrote on the genotype layer, but put circles around them to show that they're gametes, because here are the sperm and the mothers are eggs. And now we need to do the Punnett square. So mother, father. I don't know why my iPad sometimes undoes what I've done already. So she's small b, he's big B, little b. Let's cross them. So big B, small b, big B, small b, small b, small b. And therefore both of these will have blue eyes. Both of these will have brown eyes. So as a percentage likelihood, it's 50% will have blue eyes and 50% will have brown eyes. Cystic fibrosis is a recessive disease. A carrier mother and a carrier father decide to try for children. What is the probability that their child will have cystic fibrosis? So it's recessive, which means to have the disease you need this genotype, small c, small c. It doesn't matter what letter you use to assign, by the way, but I'm using C here because of cystic fibrosis. A carrier mother and a carrier father, that automatically tells me that this is their genotype, and you must learn that they're effectively heterozygous if they are carriers. So we've worked out their genotypes, so we're ready to do the genetic cross by writing mother and father at the top again. Phenotype, genotype, and gametes in the table. The phenotype is that they are both carriers. Their genotype we know is heterozygous, so it's big C, small c. This means that half of her eggs will be big C, half of them will be small c, and the same with the sperm. Make sure you really show a difference in the size of your letters here. And now it's time to do the Punnett square. So this child is big C, so they'll be healthy. This child is big C, small c, so they'll be carriers, but they'll still be healthy. Same for this one. And lastly, this child here will have cystic fibrosis. So they have a 25% chance of having a child with CF. Let's have a look at sickle cell anemia now. Now this is a pretty unpleasant disease which affects red blood cells, which, if you remember, the healthy red blood cell has this distinctive biconcave disc shape. And why does it have that shape? Well, it's to increase the surface area to volume ratio so that you can carry more oxygen on the hemoglobin. So it's all about maximising oxygen transport within the body. Now, someone with sickle cell anemia ends up with sickle-shaped red blood cells, and a sickle is like a crescent moon. These have an incredibly different shape from our concave disc shapes, and it means that much less oxygen is transported, and it can be life-threatening. So now we need to look at the inheritance of sickle cell anemia. You need to know that sickle cell anemia is a recessive disease, which means in order to have it, you need, let's assign the letter S, you need two small s's. So if you have a big S, little s, or big S, then you're going to be healthy. And if you have small s, you're going to have sickle cell. And usually a selection pressure exists, which would mean that there's a selection pressure against a particular disease to try and wipe it out from a population. However, sickle cell anemia is very interesting because in certain parts of Africa, you actually find that this particular genotype, big S, little s, is extremely good. So it's good for two reasons. First of all, because you don't have sickle cell anemia. As I've already said, you're healthy. However, it does affect your red blood cells a little, and it's been found that malaria, which is that disease which affects red blood cells, remember it's the mosquito that transmits the plasmodium, which is the parasite which causes malaria. Now, the plasmodium, which causes malaria, will not infect sickle cells. It knows there's something up with them. So that means that this big S, little s combination, where there are some changes to the red blood cells, it means that you can't get malaria. So there's a very strong selection pressure for this because malaria kills so many people. Having the genotype combination of big S, little s is actually really good, whereas having big S isn't great because although you don't get sickle cell, you're more susceptible to getting malaria. And then looking at the little s, we know it's sad because you get sickle cell anemia, but you don't get malaria. So there's a real combination going on here, whereby small s is no good because you have sickle cell anemia, but the good thing is you don't get malaria. Big S is good because you don't have sickle cell anemia, but you are susceptible to malaria. The ideal combination is big S, small s, and therefore there is a strong selection pressure for this in certain parts of Africa. So let's look at actually doing a genetic cross, which is all to do with sickle cell anemia. So we're drawing a genetic diagram to show the inheritance of sickle cell anemia in the Jones family. And Mr. and Mrs. Jones are both carriers for the disease, which means they're both big S, little s. So starting from the beginning as usual, Mr. Jones, Mrs. Jones. Always lay out your answers in this way. We're starting with the phenotype, which we know is that they're both carriers. Their genotype I've just drawn. You need to draw the alleles here, big S, little s for both. Try and distinguish clearly between your upper and lower case. The gametes, so the sperm will either be big S or little s, big S for the eggs, or little s. So let's now do crosses. So we have big S, little s, big S, little s, little s, little s, and only this child here will have sickle cell anemia. So that's 25% will have sickle cell anemia because they have little s, and 50% will be carriers with big S, little s, and 25% will be healthy with big S, and it's worth drawing the genotypes which are assigned to each phenotype. So let's now look at how we inherit our sex. And remember, this is all to do with chromosomes. So women have 2 X's and men have an X and a Y. And that is the pair of chromosomes which dictate your sex. So let's literally have a quick look as to why 50% of the population, or approximately 50%, are male, 50% are female. So let's start by writing mum and dad. As per usual, we're going to write phenotype. So what do they look like? Well, the mum's obviously female, the father is male. The genotype, you're going to use the sex chromosomes here, so she'll be XX, he'll be XY. The gametes, so obviously every egg will be X. Sperm, weirdly, can either be female or male sperm. And then if we look at the cross, let's cross the mum up here, the dad down here, and you see this situation which shows 50% are XX, so they're female, 50% are XY, so they're male. Although we know, based on that evidence, that the man's sperm determines the sex of their child, and the chromosomes, so the XX or the XY, contain lots of genes associated with the development of sex organs. You might not know that they have lots of other roles which might not be to do with sex at all, and they code for characteristics which are completely unrelated to sex. So the sex chromosomes also carry genes that code for characteristics unrelated to sex. And a very famous example of this is colour blindness. So that's whether you can see in full colour or not. And because it's carried on the X chromosome, so a sex chromosome, we say weirdly that colour blindness is sex linked. Let's look more closely at colour blindness as a sex-linked characteristics, and a very well-known one is red-green colour blindness, which is when you can't tell the difference between the colour red and the colour green. And it's carried on the X chromosome only. And when we do our crosses, just so you know, in order to have colour blindness, you need to have a small c. So if you're a guy, to have colour blindness, you need to have a X small c. The Y, you're not going to have a C on it, because remember I already told you that it's carried by the X chromosome only. As a female, to have colour blindness, you need to have 2 small c's. If you have a big C and a little c, then you're just going to be a carrier, and if you have 2 big C's, then you're going to be completely normal. So let's do a question to try and make this a bit more straightforward. So we're going to cross a carrier woman and normal man. So using our normal way of writing out our crosses, so we've got a woman and a man. The phenotype, first of all, well, we know the woman is a carrier, the man is normal. The genotype, as you look at my crosses up here, she needs to have a small c and a big C in order to be a carrier. And he's normal, which means he must have a big C attached to the X. And remember, the Y chromosome doesn't carry any genes for colour blindness, so we leave that as it is. So the gametes. The woman's eggs will either be big C or small c, and the man's sperm will either be big C or just a Y, which will obviously code for a male sperm. So now let's do the cross. So this first offspring will be just a normal offspring. The second offspring will be a carrier of colour blindness. This male offspring will be normal, and this final male offspring will have colour blindness. So let's write them out. 25% of the offspring will be male and have colour blindness, and that's due to their combination of alleles, which is X small c Y. 25% will be male and they'll be normal because they have X big C Y. 25% will be female and they'll be normal, and 25% will be female and they'll be carriers with the big C small c.
Codominance is when both alleles are expressed in an individual. The example here we use is red snapdragons.
So we've already looked at codominance, but they're very interested in the CIE exam board in how it works from a blood group point of view. So there are 4 different blood groups. They are A, B, AB and O, so you need to learn those. You also need to learn the genotypes which represent each blood group, and from a codominance point of view, remember that we have got to write them in this very specific way, which is we choose the base letter and then we have a superscript showing the allele for the particular blood group, and that will be more clear once I start drawing them out. So for A, the potential genotypes are IA, IA or IA, IO. For B, you're looking at IB, 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 IO. For AB, you're looking at IA and IB. And for O, you need IO, IO. Just make sure you're clear that that's an OR. And now let's have a look at an example so you can actually see it in practice. So let's look at an example to make it nice and straightforward. So we're drawing a genetic diagram to explain the inheritance of blood group in the Smith family. Mr. Smith has the genotype IA-IB and Mrs. Smith has the genotype IO-IO. So using my method from before, we're going to lay it out as a table. So Mr. Smith, Mrs. Smith, start with our phenotypes. So what blood groups do they have? Well, according to my previous slide, IA-IB will obviously be blood group AB for Mr. Smith. Mrs. Smith has IO-IO, which means her blood group is O. So what are their genotypes? Well, happily that's been given to us in the questions. That's just IA-IB for Mr. Smith, IO-IO for Mrs. Smith. So the gametes, we just need to separate those alleles and draw circles around them to show that this is the potential alleles for his sperm, and her eggs will all be IO. Now let's do the Punnett square. And just cross them. And then from the previous slide we know what each of the child's phenotype will be. So IO IA, that's 50% of the offspring, and they'll have blood group A. And IO-IB is the other 50% of potential offspring, and they'll have blood group B. So now we need to look at pedigree diagrams, and the best way to do this is by showing you an example. Always use this approach, and do notice these are supposed to be really difficult, so don't worry too much if you're finding it too much. Question 3. Familial hypercholesteremia FH is an inherited condition caused by a dominant allele. That is key. People with the condition have high levels of cholesterol in their blood, increasing the risk of dying from blocked arteries. The diagram shows the pattern of inheritance in several generations of a family with familial hypercholesteremia. So do notice with the pedigree diagram that the squares are always the men in the family. You'll know this from the key. The circles always represent females. And in this case from the key, we can see that the grey shaded boxes are sufferers of FH, whilst the white boxes or circles are non-sufferers. So person A is heterozygous for FH. Use this information to complete the table. So let's start by labelling the genotype of person A, and we are going to use the letter D. I mean, it doesn't matter what letter you use, but I'm going to use the letter D because you can easily see the difference between a capital D and a small d. So labelling their genotype, this is what they look like, so big D, small d. So what is the question actually asking? How many people have the genotype which is homozygous recessive? So homozygous recessive, homozygous meaning the same case, recessive meaning lower case, which is why we're looking for small d here. Homozygous dominant, homozygous meaning the same, dominant meaning that they're both capital. So that's what we're looking for. Now we're going to work out what the pedigree diagram tells us. First of all, I'm going to look at all the people without FH, so all the people that are either white circles or white squares. Because they don't have the disease, I know therefore that they are small d, so I can just label all of their genotypes straight away. And now we need to count them to work out the number of people with the genotype homozygous recessive. And once I've done that, I can see that it is 11. Now we're getting slightly more difficult by looking for the big D, so homozygous dominant. So we need to infer things from the pedigree analysis. First of all, look at woman C. So she got her genotype from parents A and B. Now B is homozygous recessive, which means they must have passed on a small d. She has the disease, which means she must have a big D, so this is her genotype. E has the same issue, but they're a man, so they're going to be big D, small d. Person G inherited a small lowercase allele from D. They have the disease, which is why they're capitalized, and the same goes for person J. And then looking at NOP, well, they inherited a small d from their mother. They have the disease, which is why they have a big D. So actually, looking for people with big D, the answer here is 0. Person G and H have 3 children, all of whom have FH. What is the probability of G and H having 3 children who all have FH? This is a crazy amount of work for 1 mark, because the only way I can see of doing this is to draw a Punnett square. So we're going to do a Punnett square for G and H using my layout I already described. So we're looking at the phenotype, genotype, and gametes. From the key, we can see that person G has FH. H is therefore healthy from the key. We've already labelled their genotypes, so we can just copy that directly across and then separate these out to see the gametes. Now just simply do a cross, and it's these 2 here that will have FH. Now what is 50% as a probability? Well, it's 0.5, and the question asks the probability of all 3 children having FH. So remember, when we're talking about probability, we have to multiply together our probabilities. Pop that into your calculator and you'll get a value which is 0.125.
Lyra! Hey! Did you come in? Oh, you're all soggy. Where isn't it focusing? You're so soggy. Is it wet out there? Super soggy. So how is variation within a species brought about? Because we know the human race isn't full of billions of people that all look the same. That is brought about by a combination of things. First of all, genetics, and secondly, environmental factors. So 2 identical twins, regardless of the fact they have the same genes, if you move them to opposite parts of the world, it's very likely they'll have different heights, different masses, slightly different skin colour, and that's due to the environment they experience. That could be lots of sun, one of them could eat more, one of them could do less exercise. You alright? Are you too muddy? You're purring, aren't you? This makes a change.
Let's look at continuous and discontinuous variation now, and hopefully you've met these two words in maths, so you already have a clue a little bit about what they mean. Now, continuous is all to do with having a range of values, and it results in a range of phenotypes which fall between two extremes. So, for example, your height. So taking people in your year, you'll have the shortest person in the year, which let's pretend they are 120 centimetres tall. The tallest person might be 200 centimetres tall. I don't know if I'm making up silly numbers, by the way. In the UK, for some reason, we describe height in terms of feet, so I'm 5 foot 4 inches, which is really silly because we use the metric system for everything else. Whereas discontinuous variation results in limited phenotypes with no intermediate values. And we've already met a really good example, which is blood groups. You're only one type of blood. So you could be A, you could be B, you could be A, B or O, but you're not a combination of all four. The same could be said with something as silly as tongue rolling. The general thinking here is that some people can roll their tongues, some people can't, and you can't kind of do it halfway. And be aware that this is all to do with genes, which you should know from doing the genetic cross.
What is a mutation? It's a rare random change to the genetic material of an organism. So a mutation can be brought about by a number of things. Things like ionizing radiation, exposure to UV light, X-ray exposure, and various mutagens, which are just chemicals which cause mutations, and you find those in things like cigarette smoke. Now, the crucial thing with mutations is what they do is they alter the DNA of an organism. We've already looked at protein synthesis, so if you think about it, if you alter the DNA of an organism, what that will do is it potentially alters the sequence in which amino acids are assembled, and therefore it can alter the end product, so the end protein which is produced. Proteins are responsible for phenotypes, so our physical appearance, so mutation can therefore cause an alteration in our phenotypes. Now, not all mutations cause this alteration, because sometimes a mutation occurs where the DNA, although it is changed, it doesn't actually alter the order in which the amino acids are assembled, so you end up with the same protein here. It's more of a common sense topic, but I do know that some of you struggle with it, so I don't want to ignore it. Right, adaptation, that is to do with organisms and them having characteristics which mean that they are better suited for a particular environment. I'm going to take it from an animal that lives in a cold environment, an animal that lives in a hot environment, and we're also going to have a look at plants. So let's just dive straight into looking at things like polar bears and Arctic foxes. Right, what you will find is, first of all, they have white fur, Y for camouflage, so they blend into the surroundings. They have something like a polar bear, I'm going to actually use that. They have large feet, Y to stop themselves sinking into the snow, because if you have larger feet, then you have a larger area to spread their weight over. It's the same reason why skis work, is because you just don't sink in as much. Right, what else do they have? They have small ears. Why? Because they need a small surface area here, because they want to minimise their heat loss to the surroundings, as obviously it's very cold and they want to keep warm. They'll have a thick layer of fat for insulation. They'll have thick fur for the same reason, to conserve heat. If they ask you stuff like, oh, adaptations for catching prey, then you need to talk about sharp teeth for tearing into flesh, long legs so they can run fast, but they don't usually ask that. It's more of adaptations to their environment. If we take an animal in a hot country, something like a camel that lives in a desert, again, they have large feet so that they don't sink into the sand. They have large ears to increase the surface area, to maximise the amount of heat that they can lose, because obviously they don't want to overheat under the hot desert sun. You'll find that they have long eyelashes, which will prevent sand getting into their eyes. They have thin fur, thin layer of fat to minimise the amount of heat that they maintain. Right, lastly, let's look at the cactus. Now, the cactus obviously again lives in the hot environment. It stores water in its stem. It has spines rather than leaves, and what that does is it prevents water loss by transpiration. They have shallow, extensive roots. I hope that makes sense to you. Basically, their roots spread out a very long way, meters and meters away from the actual plant, and what that means is if it rains, then what can happen is that the plant can absorb as much of that water, even if it's really far from them. Very short topic now on evolution and natural selection. So first of all, your definition for evolution, it states that many organisms which are alive today and many more which are now extinct first evolved from very simple life forms that first evolved over 3.2 billion years ago. So that's basically saying that evolution states that we all evolved from small life forms like bacteria, which became multicellular, which became more and more complicated. They became reptiles, they became birds, and then they became mammals, and then we came about. So that's really what evolution is stating. Natural selection links very nicely with this. Remember, this is Charles Darwin's theory. Now, he stated, and I do just want you to learn this as a 5-mark answer off by heart, he stated that there is variation within a species due to mutation, which is what I've just discussed. So within a species, there is variety. This means that some individuals within the species are more likely to survive because they are better adapted. Because they're surviving, they're likely to reproduce, so produce offspring, and those offspring will inherit those favourable genes. So before you know it, you have many generations that go past and they've all inherited this favourable gene, making them more likely to survive. And I'm now going to bring up that perfect answer for you. Natural selection can be seen pretty much everywhere on Earth, including bacteria. So we're just going to describe how bacteria may become antibiotic resistant, and it does link to natural selection. So what happens is you have a colony of bacteria, you give them an antibiotic, and due to mutations, some of those bacteria are stronger, they are resistant, that means they are not killed by the antibiotic. So what happens is all the other bacteria are killed, leaving behind these very strong antibiotic resistant bacteria. They soon replicate, and before you know it, you've got a colony of bacteria which is no longer treatable using antibiotics. And that's why everyone's so scared about antibiotic resistance and what it means for our future medicine. Now, selective breeding. So remember, this is when humans use animals or plants with desired characteristics. They force them to breed, and then they repeat this process over many generations. So before you know it, you have animals with desired characteristics. So if we're looking at animals, let's, for example, look at the dairy industry. So dairy cows, clearly a good animal here will produce a lot of milk. So humans, so make sure you point out that it's humans, they select a dairy cow that produces a high yield of milk. They mate her with a bull. It's quite hard to determine the bull because obviously they don't produce milk, but they'll mate her with a bull. And then her calves are likely to produce more milk, the female calves, because of their high yield mother. Then you take those calves and you keep repeating the process until you have lots of calves and lots of cows that produce lots of milk. And you can do the same with plants. So you can selectively breed plants to be a particular colour. So you pick flowers that are a particular colour. You force them to cross-pollinate. And then before you know, you've got a load of plants with your desired characteristics, such as petal colour. Ecology now, not my favourite topic, mainly because it's full of disgusting definitions which all seem to be very similar and all sound the same. So let's start by looking at the definition of environment. That is the total non-biological components of an ecosystem. So we're looking at the soil and the water, for example. The habitat is the place where a specific organism lives. Now, population, be very specific with your keywords here. This is all the organisms belonging to a particular species which you find within an ecosystem. What is the community? This is the population of all species found within a particular ecosystem. Now, what is a producer? Because remember, producers start all food chains and food webs. This is just a plant which photosynthesizes to produce its own food. A consumer is an animal which eats other animals or plants. What is a decomposer? It's an organism which decays dead material and helps to recycle nutrients. Define a parasite. This is an organism which lives within another organism, causing harm to that organism and feeding off of them. What is a predator? It's an animal which kills and eats another animal. Gosh, these definitions do keep coming. What is biodiversity? That's the variety of plants and animals found within an ecosystem. What are biotic factors? These are living factors. So these are living factors which affect organisms' lives. So it could be other animals competing for food, competing for nesting sites, bringing disease and pathogens to other organisms. Abiotic factors are non-living factors which affect organisms such as soil, pH, temperature of water, carbon dioxide availability, number of daylight hours, etc. Looking more closely now at pyramids of numbers and pyramids of biomass. So remember, a pyramid of numbers simply shows you the number of each organism at each trophic level. So trophic level is just the stage in a food chain. So for example, a pyramid of numbers could start with grass. The grass could be eaten by rabbits, so that will be the next tier, and lastly you'll have foxes. But we don't like using pyramids of numbers because often they end up looking a really strange shape and not being pyramidal at all, and that can be due to the producer only being one organism, such as a tree. That will be very small in comparison to the number of sparrows living on it, which is why you end up with very funny shaped pyramids. So we use pyramids of biomass because that actually shows the mass of living material available, and therefore the oak tree, for example, will appear much larger, and therefore the pyramid will be the correct shape. I'm just going to talk you through a couple of food web questions just so you know how to answer those.
The diagram shows a food web from a habitat. Use information in the food web to complete the table. The first one has been done for you. This has been drawn quite horribly, so rather than looking at where the layers are, you're going to need to count the arrows and make sure you remember things about what a producer is and what a primary consumer is. So starting with the number of organisms, which they've done for us, which is annoying because that's the easy question. Just count how many different organisms there are 8, so we do agree with them. The number of different types of plant. You're looking for the producers here. So what here is a plant? Well, you can see from the picture, even if you don't know what a cattail is, that it's a cattail and marsh grass. So the answer here is 2. The number of animals, so these are the animals which form anything from the primary, secondary, tertiary, consumer level. So the animals therefore are grasshopper, cricket, shrew, frog, snake and hawk. And if you count those up, you'll get 6. Number of primary consumers, so remember they appear straight after the producers. So we've got an arrow leading to the cricket and the grasshopper, which is why the answer here is 2. Number of food chains, God, this is more difficult. So food chain 1 will be marsh grass, grasshopper, shrew, hawk. Food chain 2 is the same but includes the snake before the hawk, so that will be the second one. Food chain 3 is the cattail, cricket, shrew, hawk. Food chain 4 will be the same but include the snake. And then food chain 5 will include cattail, cricket, frog, snake and hawk. So that was pretty tricky, but the answer here is 5. The plants in this food web make the food for some of the animals to eat. Give the name used to describe these plants. I've already mentioned that a lot. That is a producer. The hawk catches and eats its prey. Give the name used to describe the hawk in this food web. So I told you earlier in my definitions that something that catches and eats something is a predator. A pesticide can be used to kill the grasshoppers in this habitat. Describe the effect that killing grasshoppers would have on the number of shrews. So we need to have a look. Oh right, so the grasshoppers are eaten by the shrews. So clearly if we reduce the number of grasshoppers, the number of shrews will reduce because there is less food for them. Describe the effect that killing grasshoppers would have on the number of marsh grass plants. So if we have fewer grasshoppers, we can see that they won't be eating the marsh grass as much, which means that the marsh grass population will increase.
Why is so little energy passed from one organism to another? So for example, from the producer to the primary consumer. So let's take grass, the producer and a rabbit as an example. Remember, the rabbit is the primary consumer. The issue here is that only part of the grass is digestible. Much of it passes out of the rabbit as feces. Some of the grass isn't eaten, so the rabbit won't even eat the roots. I don't know if that's true, but it can be a reason. The rabbit moves, it keeps itself warm, it respires, it digests, I've already mentioned this, it poos. So these are all ways in which energy is lost within a food chain. In fact, 90% is lost at each stage of the food chain. If they ask you where all that energy originates from, remember that is the sun.
Now there's a greater push at the moment for people to become vegetarian and vegan, which means eating no animal produce whatsoever. And it's not just because it's much kinder to the animal. It's all to do with energy flow. So taking grass, and say we feed it to a cow, and then the cow is eaten by a human. Well, you know from food chains and the ecology topic that over 90% of energy is lost at each stage of the food chain. And so the more stages, the more trophic levels you have in a food chain, the more energy that will be lost. So if you can cut out that cow stage and go straight from grass to human, I know humans don't eat grass, but let's say we pick peas, for example, then we don't lose 90% of the energy at the stage before. Most of that energy will go straight from the peas to the human. So it's a much better and much more efficient way of eating, as if we all became vegetarian, and far less energy would be wasted.
Let's do the carbon cycle now. I do like this topic. If you just learn it as a list of steps, it's far easier than learning it as the whole cycle, unless you're a very artistic kind of pictorial person, because I really struggle in that way, but I find learning this list of steps works well every time. So we're looking at how carbon is cycled in our atmosphere and within living organisms. So the place I like to always start is carbon dioxide in the air. So what happens to that carbon dioxide in the air? Well, it gets absorbed by green plants in photosynthesis, and it is used to make glucose. Those green plants then respire because they're living organisms, and that releases carbon dioxide back into the atmosphere. So here's the first step of CO2 moving in, CO2 moving out. The plants are eaten by animals, and so that carbon that was part of the plants becomes part of the animal body, and then the animal respires again, releasing carbon dioxide into the atmosphere. Lastly, plants and animals inevitably die, and then this is where decomposers come in. They break down that dead material and they respire, again releasing carbon dioxide. So we can see carbon dioxide went in the first place with green plants photosynthesizing, and then it left via respiration by plants, animals and microorganisms. Do notice that combustion, which is burning of fuels and things, also releases carbon dioxide into the air. Now the nitrogen cycle, again start in one place and allow your answer to feed in from there. So I like to start with nitrates in the soil. So remember, those nitrates get absorbed by active transport into the root hair cells of the plants. Nitrates are important in the plant manufacture of proteins. So the nitrogen in the air becomes locked up in proteins within the plant. The plant is eaten by animals, and therefore the nitrogen moves from the plant into the animal. And then both the plants and animals die and microorganisms become important again. So the ones you need to know about are nitrifying bacteria, and they convert the ammonium within the dead matter from nitrites to nitrates. So nitrates have been returned to the soil, nitrogen has been returned to the soil. Do notice there are some really annoying bacteria called denitrifying bacteria, and what they do is they take nitrates in the soil and convert it back to nitrogen in the air. So they're not very popular with farmers, because after all, the farmer wants lots of nitrates in the soil. They don't want nitrogen gas being released into the air because there's plenty of nitrogen in the air. So that's why denitrifying bacteria are particularly frustrating. The last thing to notice is nitrogen fixing bacteria, which we find on root nodules, so small bumps on the roots of leguminous plants. Legumes are a family of plants which include peas, beans and clover. So they're super good to plant because they provide a lot of nutrients to the soil. The reason being that the root nodules containing nitrogen fixing bacteria, they convert nitrogen gas in the air to nitrates. So effectively they make the plant its own supply of fertilizers. So that's really great, because by planting clover or peas or beans, you're basically fertilizing the soil. So say the farmer regularly plants wheat as their cash crop, they might choose to crop rotate and plant clover every 4 years just to add a nice input of nitrates to the soil.
So now let's look at the factors which affect population growth. We're going to use an example of cows. So there's a population of cows and they want to increase their number. So what factors would affect how quickly they can increase their numbers? Well, first of all, food supply. If there's not much grass available, then there isn't a lot of food for them to eat, so obviously they won't be able to grow and they will starve. Predation is a super key point. If there are lions around and the lions eat the cows, I'm talking about in Africa, by the way, I hope you don't think I'm sounding really silly with this example, then clearly that will limit population growth because you have fewer cows because they have been killed and eaten by the lions. And disease is a very important factor too. Other cows might introduce pathogens to our original cow population. The lions themselves could introduce pathogens. So disease has always been a huge way in which large numbers of people or animals have been killed. So for example, malaria has been responsible for millions of human deaths. The same is true for typhoid, cholera, tuberculosis, etc. However, more recently, these diseases have been under control when we're concerned with the growth of the human population. So over the last 250 years, if you have a time on the x-axis, human population on the y-axis, and we've seen it just go extremely high in just the last 50 years alone, and that's all to do with things that we've just discussed. So disease, medicine has meant that many people who would have previously died are surviving. There's improvements in water supply and cleanliness, really, so not just having more water but making sure it's clean. To do with that is sanitation, so making sure that our waste is dealt with appropriately and cleanly, and we have better food supply. However, this astronomical increase in human population has huge environmental and social implications. It's led to global warming due to the enhanced greenhouse effect, and that's all due to burning fossil fuels. Deforestation is going on an awful lot, cutting down trees to make more space for animals to feed, also to give us building materials for our houses. And we're seeing an ever increasing destruction of the ozone layer due to CFCs in our fridges and aerosols. So these are all impacts of a massive increase in our population.
Right, looking at microorganisms involved in food production, so primarily yeast. So how is yeast used in bread making? Don't forget that yeast is a fungus. When it's forced to respire anaerobically, it breaks down glucose, or it respires glucose into carbon dioxide and ethanol. That carbon dioxide is very important in bread making. It creates bubbles, which actually helps that bread dough to rise. Looking at the role of yeast in beer making, so the anaerobic respiration of yeast, remember that produces ethanol, I've just told you that, and ethanol is an alcohol, so that's where the alcohol found in beer originates. Moving on to fermenters. So what is a fermenter? Well, it's a vessel which contains microorganisms which are involved in fermentation reactions. So let's describe the structure of a fermenter and how it is optimized to ensure as much of the product is produced as possible. So first of all, we control the temperature, and that's through a cooling jacket, because microorganisms, when they respire, produce a lot of heat, and if too much heat is produced, then they can denature the enzymes and actually kill themselves. So the cooling jacket has cold water flowing around it, which helps to remove excess heat from the fermenter. Coupled with this, you have temperature and pH monitors, because obviously you need to determine if the temperature is too high, you need to determine if the pH is too alkali or acidic. So it's important that we keep watch on the fermenter. We also have stirring paddles to mix up the contents, and that ensures that the nutrients and heat are evenly distributed. You often have an air inlet, not always, but sometimes, and that's to allow oxygen into the fermenter for any microorganisms which respire aerobically. And lastly, nutrient supply, because obviously the microorganisms need something to respire. Genetic engineering now. This is quite a complicated topic. It is chock-a-block full of key scientific words, but if you learn them off by heart, you should be fine. So remember, we genetically engineer things like insulin. So insulin is a hormone produced by our pancreas, and it's responsible for lowering our blood sugar levels after we've eaten. And for type 1 diabetics, they find that they don't produce insulin, so they really struggle to maintain their blood sugar levels, which is where genetic engineering comes in. Because in the olden days, they used to obtain pig insulin, so they used to chop into pigs, remove the insulin, and not only do you have major ethical issues with this, but obviously the insulin wasn't particularly fit for purpose because it came from pigs. So it was important that we found a way of producing insulin from humans, and so that's where genetic engineering came in. And when we're talking about genetic engineering, we're talking about using bacterial cells, because bacterial cells contain small rings of genetic information called plasmids, which we can manipulate so that you can insert the insulin gene and force the bacteria to produce lots of insulin. So let's go into great detail how that is done. So we obtain the bacterial cell and we cut open the plasmid using a restriction enzyme, which acts as a pair of biological scissors. Then we use a restriction enzyme to cut the insulin gene away from the rest of the cell, and we insert that insulin gene into that bacterial plasmid using a ligase enzyme. And we stick it together, and that's why we say it has sticky ends. Once we've done that, we're ready to put the bacterial cell into a fermenter, and it's been done many times. And then I've already mentioned the fermenter. You need to provide it with the optimum conditions, so the right temperature, the right pH, the optimum amount of oxygen and nutrients, etc. And before you know it, your bacteria has made huge amounts of insulin. Don't forget a few keywords here concerning genetic engineering. Once that plasmid has a different gene inserted into it, we call it a recombinant plasmid, which means it's been recombined, so it's been changed. And don't forget also that the bacterial cell, the plasmid, is acting as a vector, which means it transports biological material from one place to another. We can also genetically modify plants so that they can have desired characteristics. This could include being frost resistant, so that stops them dying when frost hits in winter. It could be to extend their shelf life to stop them going off so that they have a longer shelf life and are more fit for human consumption after many days. You might actually want to make plants resistant to weed killers. This sounds really strange, because why would you want to make a plant resistant to weed killer? But think about it. A farmer applies the weed killer. By the way, the best name for a weed killer is herbicide. Side meaning kill, herb meaning to do with plants. So the farmer applies the herbicide. He or she wants to kill the weed. However, some of it will inevitably fall on the plant that they're trying to grow. And obviously you don't want that to happen, because it will actually kill the plant you're trying to grow. So if you can make that crop plant resistant to herbicide, then that's great, because you apply that herbicide or weed killer, it kills the weeds, and your actual plant that you're after stays alive and continues to grow. You can also genetically modify plants so that they can actually have health benefits. One of the most famous examples of this is golden rice. So when poorer countries grow lots of rice, unfortunately rice doesn't have a huge amount of nutritional value. So what you can do is genetically modify it so that it contains vitamin A, and therefore when people eat that rice, they get a huge amount of vitamin A in their diet, and that stops them getting night blindness. This is something you should remember from the balanced diet topic of the specification. So golden rice is an excellent example where genetic modification has been used really well. And then the most strange example of genetic modification is when we talk about tobacco plants, and these have been modified so they actually produce hepatitis antigens, therefore have a potential vaccine against hepatitis. So this is like crazy science, but just remember that tobacco plants may be modified to produce hepatitis vaccines. So we've already touched on this, we've given lots of examples of genetic modification in plants. Just to reiterate the advantages and to throw in a few more, you can have increased salt tolerance,
The expectation is that the human population will reach an incredible 9 billion people by the end of this century. And that is crazy. 9 billion people need feeding. So how have we managed to provide so much food? How are farms generating so much food, both from an animal and crop point of view? So let's look at these different factors. So we've improved productivity by using machinery, So in the olden days when they used to use ox and plough, increasingly modern technology sees us using machinery, and what that can actually do is help remove hedges. And if you remove hedges, then you have larger fields and therefore more space to grow crops and animals. A large part of productivity to do with growing crops is how fertile the soil is. So we can increase the soil's fertility by using fertilizers, often NPK ones, which remember contain lots of nitrogen, phosphorus and potassium, which are all needed to encourage healthy root growth, prevent yellow spots, help build plant proteins, etc. Coupled with this is the use of pesticides. And this includes both insecticides, so killing insect pests which feed upon our crops, as well as herbicides, which are weeds effectively that compete for nitrates in the soil, for sunlight, for water, etc. So if we can kill the weeds and we can kill the insects which feed upon our crops, we can increase the amount of crops we produce. And then selective breeding, which is all about humans picking animals and plants with desirable characteristics. And what that means is the end result is better. So from an animal point of view, that could include picking cows, which are good milkers, so produce lots of milk, and you keep crossbreeding good milking cows with bulls, and then eventually you end up with a whole population of cows which produce loads of milk, which is obviously great for the farmer. From a plant point of view, it could mean picking plants which are resistant to disease. Maybe they're hardier, so they don't get killed by frosts, which is a big problem in the colder countries. And then the biggest reason for improved efficiency is monoculture. So historically, farms were small and mixed, so they contained various animals and crops and plants. Monoculture is the tendency to pick a single crop. So let's look at both the advantages and disadvantages of monoculture, because with all of these things, there are always good and bad points. So starting with the good points, you can selectively breed plants. So what I was just saying, you can selectively breed plants with ideal characteristics. And you can match them to the type of soil you have to make sure you're maximizing growth. And because you know your plant so well, you know its exact mineral and water needs. So you know to avoid deficiency diseases. And then lastly, in terms of the machinery you use, if you buy a very specific kind of tractor for a particular type of wheat, then you know it can quickly harvest. And we call this specialised harvesting techniques. However, with the good comes many bad points. And as is the way when you grow lots of the same plants close together, you increase the chance of disease. And a disease that can particularly affect plants is the tobacco mosaic virus. And that can easily spread from one plant to the next. Because you've decreased the biodiversity, so you've decreased how many different types of plant there are, you end up affecting the wildlife, as there's less food for birds, less food for butterflies, etc., etc. As you often end up with fields containing the same type of plant, This leads to a loss of genetic variation. And remember, with this comes problems with natural selection, because if the conditions change, you find that you don't have a population of plants with enough genetic variation, so they're more likely to die out. And lastly, you see damage to the soil as the minerals get leached away due to the same plants being present. We've already said that we can remove hedges and create larger fields, and that's due to specialised machinery. And now we need to look more closely at that and look at the advantages and disadvantages of the removal of hedges. And these should be nice and obvious. So the most obvious reason you would remove a hedge is to provide more space to grow crops. Another obvious point is that hedges provide shade, and where there's shade, you're going to have less light, so less photosynthesis by your crop plant. Hedges often provide shelter for animals such as rabbits, and rabbits are very destructive forces as far as the farmer is concerned. Their burrows destroy land, and the rabbits eat the crops. Again, a big no-no from the farmer's point of view. And then lastly, they often need trimming, redigging, replanting, so they need a lot of labour in order to maintain them. Disadvantage then, so what are the bad things concerned with removing hedges? These are mostly environmental. So first of all, hedges act as windbreaks. So without hedges, you have no windbreaks, which leads to the erosion of soil. Increasing the leaching of nutrients. Hedges also provide a great ecosystem. Hedges also provide a great habitat for many animals, for many bird species, so we lose nesting sites. And coupled with that is no shelter for small mammals. And then lastly, the leaves which fall from the hedges may be cycled into the soil, adding nutrients. So we have no nutrient cycling if we remove our hedges. Now we need to look at malnutrition and famine. Remember, malnutrition is a condition due to a diet which is low in all sorts of key important food nutrients. Lack of a balanced diet containing proteins, vitamins, and carbohydrates. If you have long periods of time where you are malnourished, this can lead to deficiency diseases. And we've met some of these in the balanced diet topic, such as scurvy, marasmus, kwashiorkor, and these are respectively due to a lack of vitamin C from scurvy. Marasmus is protein deficiency, as well as kwashiorkor. These are all due to lack of protein. So it's not just enough that people who have the deficiency disease are malnourished, that they're unwell. There are knock-on effects of them being ill, and that includes the fact that when you're malnourished, you're often unable to work. So you have no money in order to actually improve your situation to buy food. If children are malnourished, then parents often have to stay at home to look after them. Meaning that they can't go out and work, so the problem increases. And then a difficult point to remember is that older people who are malnourished are more likely to die, and they won't be able to pass on their knowledge to to younger generations. So looking more at famine, we now need to discuss the reasons for it, and the number one reason is our increasing population. Clearly means that we need more food as a population, and there just aren't enough food stores to actually meet our demands. Secondly, environmental problems relating to drought, which is lack of water. Without water, our crops die, so we have less available plant material to eat, and it also means that our animals die, because clearly animals need to drink water. Without water, they die. So we have less potential food to eat from both an animal and crop point of view. And again, coupled with water and weather is flooding. With increasing environmental change, you find that flooding can wash away crops as well as fertile soil, leaving behind nothing and no nutrients to grow new crops on. Fourth, we have the unequal distribution of food. So there are lots of places in the world which are generating loads of food, much more than the actual population which lives there requires, but it's been very difficult to actually transport this food to places where they're not producing enough of their own food. And remember, food goes off quickly. And then lastly, the high cost of fertilizers means that certain places can't afford these expensive chemicals, and therefore they can't improve the quality of their soil enough to grow their crops.
So how does using fertilizers increase crop yield? Well, the addition of fertilizers to the soil replaces leached or lost nitrates and mineral ions from the soil, because remember, fertilizers are very rich in nitrogen, nitrates, and those nitrates are used by plants to build proteins. What is a pesticide? Remember, it is a chemical which kills pests, so anything which feeds off plants will be counted as a pest. Killing pests obviously reduces their damage to the crop, and it also helps to increase crop yield. Issues though, there's lots of issues with using pesticides. Firstly, that they can be very expensive. They're persistent, which means it takes a while for them to decompose, so once you apply them to your soil, you've got to be aware that they may hang around for many, many years. And the problem here is that they can often kill animals which aren't even pests, which is really, really bad, because these are innocent animals getting killed by the pesticides because the pesticide does not discriminate correctly. So what happens here is it kills other animals. Some of these animals get eaten by larger animals, so we're talking about food chains here. And this is called bioaccumulation, where the pesticides become stored in these animals, and then as this pesticide works itself up the food chain, we call this by magnification. And the famous case study of this is DDT, which was used to eradicate malaria and typhoid in the Second World War, and there are still areas of the world where DDT is killing huge amounts of flora and fauna, and that just means animals and plants. Let's now look at human impact on the environment, and we're going to start with eutrophication, which is an effect brought about when farmers use too much fertilizer on their land and when sewage, this is disgusting, washes into rivers and streams and lakes. So do remember, fertilizers and sewage contain a lot of nitrates. So what happens is the plants use those nitrates to build proteins, and they grow extremely quickly. Because they've grown so quickly, they end up dying. The reason for this is due to lack of light. Basically, they block all the light for each other, and they die because they can't photosynthesize. My notes have gone. The death of the plants obviously provides lots of food for decomposers and microorganisms. So they grow hugely in number because they're feeding on this dead material. Because they're aerobic respirers, they use up all the oxygen in the watercourses, and this means there's no oxygen available for aquatic animals, and they die, and that's how leaching of nutrients, of fertilizers and sewage can end up with death of all aquatic animals within watercourses.
So remember, a non-biodegradable plastic is a big problem, and that's because the non-biodegradability of it means that they can't be broken down using microorganisms, because biodegradable means breaking down using microorganisms. And these sit in our landfill for hundreds, if not thousands of years, And you need to look at their issues both on land, which is terrestrial, and also in our oceans, which we call aquatic. So what issues might arise by non-biodegradable plastics being deposited here? So terrestrially, believe it or not, the plastics can block water channels, meaning that the water can't drain away and sits in the soil, which we call waterlogging. And if there's too much water in the soil, it actually leads to a decrease in the amount of oxygen available in the soil, thus decreasing the soil's fertility, meaning that plants can't grow as well. Both on land and in water, they can be consumed by animals. And if that ends up in the animal's stomach, it won't move anywhere, meaning that the animal can't eat any longer, so they're liable to starve. Going into more detail about their deposition in landfill, weirdly, when plastics are placed into rubbish heaps, so in landfill sites, they actually block the passage of oxygen. And oxygen is often needed by bacteria to break down waste, so it actually inhibits the breakdown of other waste. And lastly, if we try and get rid of them by burning them, you often find with incomplete combustion that soot is produced. And if that soot is deposited in the air, it can really affect both our and other animals' breathing.
The greenhouse effect now. The greenhouse effect is very famous. We're always talking about it because of environmental change. Do remember this is due to human activity, or we think it's due to human activity. So burning of fossil fuels, which releases carbon dioxide. Other greenhouse gases include methane. Do remember sources of methane. So some of that comes from the digestion of bovine animals such as cows. Effectively, when they fart and burp, that's disgusting, they release lots of methane. Rice paddy fields, the microorganisms that are found in rice paddy fields contribute an awful lot of methane to the atmosphere. And remember, the other two greenhouse gases you need to know about are water vapour and nitrous oxide. So there are 4 greenhouse gases: carbon dioxide, methane, nitrous oxide and water vapour. So what effect does this increase in greenhouse gas have on the environment? What you find is that the whole of the Earth's atmosphere heats up, and this leads to widespread melting of the polar ice caps. This means much more water is added to our seas and oceans, and consequently you get a rise in sea level. This floods low-lying land, so towns and cities close to the coast, and it will automatically lead to loss of biodiversity because the animals have less habitat, less places to live in, and you can see extinction of some species. We also are seeing a knock-on effect with extreme weather, huge storms, typhoons, etc. This is all a result of enhanced greenhouse effect and global warming. Acid rain, you need to know about in GCSE chemistry. Remember, it originates because of sulfur impurities in petrol, which when burnt release sulfur dioxide into the air. The high temperatures found in car engines cause the nitrogen and oxygen in air to react, again forming nitrogen oxides. So nitric acid and sulfuric acid is produced, which are components of acid rain. So what effect does acid rain have on the environment? Well, first of all, it damages trees and it can literally dissolve away their leaves. It damages limestone buildings, and lastly it gets into lakes and rivers and makes them too acidic. Deforestation, so geographers, you ought to know a lot about this. Remember, this is the cutting down of trees. Why do we deforest? Well, it provides more land for animal rearing, so farms. It provides more land for crops to be grown. It provides building materials used in houses. And that's everything I'm going to say because I can't think of anything else. And then what effect does deforestation have on the environment? Well, it's got some horrendous effects, and you can talk quite in depth about these effects. Firstly, by cutting down trees, what you're doing is you're releasing an awful lot of carbon dioxide into the air, which is obviously going to enhance the greenhouse effect and contribute to global warming. Cutting down trees means that animals' habitats are destroyed. There's nowhere for them to nest anymore, if we're talking about birds, or for animals to hide in the undergrowth if we clear the land. You get leaching of nutrients because remember those tree roots hold those nutrients, those nitrates in place. Without the trees you end up with the rain falling on the ground, washing away the nutrients into streams and rivers, meaning that the whole land remaining is barren and infertile. And tied to this is soil erosion, where because of the rain falling you get widespread flooding and landslides. And really thinking about it, the whole of the water cycle is disturbed because there's less transpiration occurring, which means because less transpiration is occurring due to lack of leaves, meaning that less clouds form, less rain, and you get a whole disruption to the weather found over forested areas.
We now need to look at the conservation topic. It's not difficult, but it is a matter of rote learning many things, including the definition of a sustainable resource. So the crucial thing about a sustainable resource is that it doesn't run out, as the name suggests. And what that really means is that this resource is produced as rapidly as it is used. Why is there therefore a push to conserve fossil fuels? And that's because of the manner in which fossil fuels are made. Remember, they take millions of years to make. They come from the bodies of dead sea animals and plants, which required a huge amount of pressure and high temperature. And because of these unique conditions needed and the time frame, they're just going to run out. We burn them, they will run out and they will not be replaced. So crucially, they run out and they are non-renewable. Looking at resources which can be maintained, this sounds very strange, but clearly resources such as forests, where we get wood from, which is building material, and fish stocks are things which can easily be maintained and controlled. Fish grow nice and quickly, they can be fed very easily as long as it's a high protein diet, and you can produce lots of fish in this way. More random information you need to know is to do with the names of materials which can be easily reused, so used again and again, or recycled, which means putting them through a manufacturing process that enables them to be used again in a different form. And such materials include paper, glass, plastic, and metal. Now we need to look at how our dirty water may be made safe. So remember, this dirty water, if it contains human waste, is known as sewage, and that needs to be taken to a treatment plant where it is later recycled, and then that water can be used again once it's been cleaned, filtered, had chlorine added to it to kill the pathogens. Why do organisms become extinct, which means the permanent loss of a particular species, or at least become endangered, which is when there's a huge loss in numbers of a particular species? Well, that's due to all sorts of things. So one thing that's very hot at the moment in the news is climate change. So when we talk about the greenhouse effect being enhanced, and we talk about global warming and the melting of polar ice caps, remember that causes loss of habitat for polar bears, etc., leading to their possible extinction if they've got nowhere to live. Other forms of habitat destruction include deforestation, which I've already mentioned, which is the chopping down of trees. Many animals make their homes within forest. If you cut down the forest, there'll be nowhere for them to live. And more actively, if we hunt animals to kill for either their flesh or their furs, that's obviously going to lead to potential extinctions. General pollution, so as we burn more fossil fuels, more acid rain, is produced, which can damage trees, again damaging the habitats of these animals. Things like lichen, very specific species of lichen. If the SO2 levels, so sulfur dioxide levels, are too high, certain types of lichen can't grow. And lastly, if you introduce foreign species, and in Britain there's been a real issue with grey squirrels, which have come over from America, and effectively they've outcompeted our native red squirrels, leading to them becoming an endangered species. So we've talked about the miserable side, which is to do with why animals become extinct, but how can we conserve endangered species? So how can we actually improve the numbers of these endangered species? First of all, by monitoring them, by providing protection for them, so whether that's giving them nesting sites or helping prevent predation. We can educate people as to their behaviour to prevent them actually leading to the endangerment of these animals. Something which is more active is a captive breeding programme where we capture these endangered animals, we give them a nice environment to live in, lots of food, so they increase the number, and then we release them back into the environment. And if we're talking about saving plants, we have seed banks where we store large numbers of plant seed for growth at a later time. Another definition you need to be aware of is sustainable development. And this is all to do with providing for the needs of the human population without harming the environment. How can fish stocks and forests be sustained? So we already talked about them as potential sustainable resources, but they're not going to sustain themselves. How are we actually going to increase the number of trees within our forests, increase fish stocks? Well, clearly by restocking, so that means adding more fish to the original amount. Educating people is always a good answer. So to prevent overfishing, to prevent deforestation, and providing quotas, which is all to do with controlling the amount that people are allowed to either cut down trees or to fish. The next thing we need to look at is why species are so at risk if their population drops. Why does that ultimately sometimes lead to their extinction? And that's because if you decrease the population, clearly you have fewer individuals, which means when they breed, you have less chance of genetic variation. And remember, that's crucial for evolution and natural selection to occur. So if you have less chance of genetic variation, then it means you're less likely to have offspring which can survive in adverse conditions. And as such, you get interbreeding, and fewer healthy offspring are produced. So we're very interested in conservation programs. Why do we carry out conservation? It's pretty obvious. It's to prevent extinction of species. Like I said, not a difficult topic, just lots to learn. It's so that we can protect vulnerable environments. So our ecosystems are maintained. Our nutrients are cycled appropriately within the carbon and nitrogen cycles, for example. And lastly, remember, we get loads of our pharmaceutical drugs come from our forests, from our tropical rainforests. If we cut them all down, we're not actually going to be able to produce our drugs that we need. Similarly, food and also our fuels, because remember, biofuels come from cutting down trees. So we'll just call that generally resource provision.
Right, I hope you found my video helpful, guys. These are so difficult to make, but I know you guys really like to just sit and watch the whole thing in one. So please give me a like, like this video if you found it helpful. It is a good incentive for me to continue my work, and don't forget to sub. I'll be back soon with another video. Bye!